Hello everyone, and welcome to the second day of the 8th European Tay-Sachs and Sandhoff Family Conference. Great to be here with you.
Hello, hola a todos. Dan, I don't know if everybody can hear us because I have some problems with the audio. Okay, I go ahead. Hola a todos, buenos días. Arrancamos nuestro segundo día de conferencia virtual este año, así que esperemos que disfrutéis. Tenemos una agenda muy interesante para hablar de los ensayos que hay en marcha, así que bienvenidos a todos.
Herzlich willkommen zur 8. Europäischen Tay-Sachs and Sandhoff Familienkonferenz, die erste virtuelle. Es ist heute ein sehr spannendes Programm und wir wünschen euch allen viel Spaß.
That's great. Like we said, we're really pleased to be here on the second day, and we have a very exciting list of speakers. And before we get going, I just want to say, for a bit of housekeeping, if you have any questions or comments you want to add, please just drop those into the stage section of the chat box. That means we can then display them into the main speakers, such as here. And it just means that people will be able to ask questions and we can respond to them very easily. So now I'm going to hand over to Brigitte, who's going to introduce today.
So, hi all, welcome to the second day of this virtual Tay-Sachs and Sandhoff conference. My name is Birgit, I'm co-founder and vice president of the German Tay-Sachs and Sandhoff patient organization. My husband and I founded the association about 7 years ago, 8 months before our son was diagnosed with Sandhoff disease at the age of 4. He's now 12 years old. We care for about 40 affected families in the German-speaking area of Europe, so Germany, Austria, Switzerland, and a part of Italy. And since this year, we care for GM1 too. The main topic of the second day of this conference are the current studies. We held our German family conference about 2 months ago with 21 affected families. We were happy to meet each other in person. But I totally understand that this year it wasn't possible to realize a personal European conference. So I'm grateful that Dan organized this virtual meeting for the English-speaking community. I heard most of the speeches of today also at our conference and in our mother tongue. I'm very happy to see the results of the last years, and I'm happy about the plans for the next years. When we got the diagnosis for our son, there was no medication at all. We could try miglustat, Zaveska, but only as off-label use. It was the first time we heard the word off-label use. Now, only a few years later, first clinical trials have started for juveniles and adults with oral drugs. And also first gene therapy trials are in progress. Other pharmaceutical industries are planning further studies. Therefore, there will be new questions for our families. The first families with affected juveniles or adults already have to take the difficult decision which trial would be the best for their child, or will there be a better one available in the next months? Can a family take this decision? It is more and more important to have neutral researchers. It will be important to develop biomarkers and to have natural history studies. And there are more questions. Will we have enough patients for all these trials? If not, how can we find those patients? Most countries in Europe have no patient organization. How can we help these families? How can we get in contact with these families? And who are the right patients for the studies? Who can judge it? Does it make sense at all to perform a study with placebo for such a rare and fatal disease? And is it ethically acceptable? For all these questions, it is important that pharmaceutical industry, researchers, expert and patient organizations work together. And I'm glad that first pharmaceutical companies came to us to work together. Hopefully, others will follow. We discussed at our German conference about the possibility of a round table with all pharmaceutical industries, researchers, and patient organizations. But at first, let's hear about the current status of the research. Today, we'll hear about gene therapy, substrate reduction therapy, and small molecules, and the difference between substrate reduction and small molecules. We hear about the importance of natural history studies and patient data. I hope we can meet each other next year in person. The presentations are important, but the talks in the evening are very valuable too. So now let's start with our first speaker, and it's a big honor and emotional moment to announce Professor Timothy Cox from the Oxford University as the first speaker. When we got the diagnosis for our son, our experts got in touch with him to discuss options for our son, because he is one of the best experts for lysosomal storage diseases. 1 year after our diagnosis, we met him the first time in person on our first ETSCC conference in Paris. Unfortunately, today only virtual, but I'm excited about your speech about substrate reduction therapies. Apart from the gene therapy, this is a big hope in the fight against GM2. Welcome, Tim.
Oh, thank you, Birgitte. I hope you can hear me. Can you, everyone? Is it all right, Dan? Can you hear me?
Yes, we can, Tim.
Oh, thank you very much. Sorry for that. Slight technical last century person from me. It's a great honor, and I wouldn't miss the meeting for anything. And still active and very much looking forward to hearing all the talks today. Afraid I was doing other things yesterday, but very much with you. Sorry about the appearance, but I will try to do my best. Dan, are you able to bring up the first slide?
I'm just adding that in, so give me 1 minute.
So sorry, it's a large.
That's fine.
It's quite a large, and I hope I'll be able to see it myself. That would help. Sorry for this technical problem.
No, that's fine, Tim. We're just going to upload. Maybe it'd be quite good for some people to hear a bit more about you.
Well, yes,
briefly
yes,
for
of course.
a minute while it uploads.
Of course, it's a 5 megabyte PDF, so it's at the limit. So, folks, yes, thank you. I worked on lysosomal diseases, and particularly this family of lysosomal diseases: Fabry disease, Gaucher disease, and GM2 for many, many years, perhaps 35 years. But I work in Cambridge, actually. Brigitte, it's very kind of you to put me in the other place. But I work in Cambridge University now, and was in London before for many years. So I work particularly on this field at the moment. And my colleague Begonia Cachon and I and other colleagues have worked for quite a few years at preclinical levels of corrective therapy for various conditions, Krabbe disease, but particularly Tay-Sachs and Sandhoff diseases. And we are still trying to raise funds for our work, which we did at one point, which was somehow went down when our great American colleagues have overtaken us. And we're very glad that there are trials happening at the moment, which we'll hear more about. Whatever will help the field is important, very important. But I also worked on substrate reduction therapy and enzyme therapy for Gaucher disease, with the view that it would apply to many conditions in this field. And so it's proved to be, actually. And what's very thrilling is that the commercial sector, which I was always unfamiliar with, is now engaged in the field. And that means that there's interest in producing orphan drugs, and there is interest in doing good things, because they won't sell unless they work. There we are. Thank you so much, Dan. I apologize for the problem. So how substrate reduction therapy is meant to work, and how it does work. It does actually work. And I'm trying to pray that it will work as we believe it will in Tay-Sachs and Sandhoff diseases. So I can talk about the principle. The next slide, Dan, will just tell you the key discovery, really. These are Gaucher cells, which occur in the bone marrow on the left in blue, and in the spleen, in the next one along. You can see a cell there, very nice color, but when you look under the electron microscope, you can see material inside these spaces, and that's inside the lysosome. That's glucosylceramide. And it's very, very closely related. In fact, it's the starting place when the body makes GM2 ganglioside, which is the material that accumulates in the brain, mostly in the brain in Tay-Sachs disease and Sandhoff disease. And you can see those are lysosomes, black there, in a neuron in the brain of a child with Tay-Sachs disease. The next slide. So the question is, how do you get rid of it? And for you, Birgit, and for, of course, your son, and I hope he's going to get better very soon after the initial injection booster. But let's hope that this discovery brings British and German and European scientists still very much together, because the discovery of the critical compounds was done by what we call the first British biochemist, and he was brilliant, but he was actually a German. And a wonderful man he was too. His last years, 40 years, was spent in the UK, and he discovered these substances, sphingolipids, in the brain. And it was brilliant work. No one ever believed him, but for me, he's a hero. Next slide, please. So these are what they are. This is the chemical compound, and these three sphingolipids here, the top one is glucosylceramide. That accumulates in Gaucher disease because it can't be digested efficiently. Underneath it is a very similar one, which is GB globoside. That accumulates in a condition called Fabry disease. Fabry was a German. Anderson also discovered it, was a Brit. And that disease, of course, is perhaps the most common of the lysosomal diseases. And then underneath it is GM2 ganglioside, which you can see has just got a few more branches on it. They're very, very similar. And these are sphingolipids. Next slide, please. They're a common family, and they all start from the one that accumulates in Gaucher disease. It's an autosomal recessive condition. It's very rare. It was thought only to affect the abdomen, the spleen, and the bones. It's quite disabling, very disabling, untreated. And it means that there isn't the turnover of a sphingolipid. As time has gone on, once we realize it affects people all over the world, we realize it's not only the liver and spleen that's involved and the bones there you see in Swedish and Polish patients, but in the little girl you can see on the right, she's also got a brain problem. And so it's very similar. And there's an excess of this material, sphingolipid, it's called, in the body and in the brain in that little child. Next one, please. So it's very similar. And here we have the adult and the juvenile forms of, and the infantile forms of Tay-Sachs and Sandhoff disease, very similar. And we all know it's the same phenomenon. Very severe mutations give rise to an infantile variant, whereas less affected mutations give rise to a more slow disease. And there are adults with Tay-Sachs disease and Sandhoff disease. So this is the one where we think substrate reduction therapy will be particularly helpful. And I'll try and show you why. Next slide, please. So this is Fabry disease, affects the brain and also the body. So that's a big market. There are enzyme therapies for that, as there are for Gaucher disease. And that's where the competition has proved to be successful for doing things in GM2 gangliosidosis, Tay-Sachs and Sandhoff diseases. Next one, please. So this is what happens. You can't digest the glucosylceramides in Gaucher disease, and so there's a problem. They build up. But the glucosylceramides are the basis for lots of these sphingolipid compounds, gangliosides and globosides. They share it very much. Glucosylceramide is the first one. GM2 gangliosidosis is about the third one. Globicide is about the third one. In the process of turning these chemicals over in the body, which have extreme importance in the body in various organs, many, many functions, but they all share a common beginner, a first compound, and it's that one. Next slide, please. So I know my slides are a bit complicated, but I'm trying to put it in context. So what is the basis for doing this? And actually, there is a piece of information from the past and a very successful treatment. This is for people who have too much cholesterol in the blood. And we know that there are prescriptions of about 7 billion prescriptions a year for the drugs called the statins, which slightly reduce the formation of cholesterol in the liver, where cholesterol is made. You don't eat cholesterol, mostly you make it. And when you make too much of it, for the rate of recycling of it, it accumulates in your arteries, and this is the cause of strokes and heart attacks and things with the poor arterial supply to the blood. So the statins slightly reduce the formation of cholesterol in the liver, and this was recognized by a Japanese scientist, Akira Endo. And when they do that, the whole thing corrects. The body readjusts, and the turnover of the cholesterol goes down, and the deposits in the blood vessels slowly get better, even in the very extreme forms of this disease, 1 in a million with homozygous hypercholesterolemia. But much more common, 1 in 500 people, is the heterozygous form, and other forms in the population anyway. These drugs work. They just slightly reduce the formation, and the body catches up. Next slide, please. And so it is with the lysosome. And this is the work of the man who discovered this, was a man in the University of Michigan. His name, who thought about this, was Norman Radin. And his pupil, his student, was Dr. Jim Shaman, and he's been very important in the field. So the idea here, this is a drawing used by Francis Platt, a very good friend who, as you know, is active in our field, and we're colleagues. She's in Oxford and I am in Cambridge, and so it will remain, but wonderful. So she worked out, and others worked out, that she had a chemical that might serve as a kind of statin. On the left, you can see a tap running. That's the input of sphingolipids into the lysosome, which is the sink or tank there. And the turnover is what runs out in the body all the time, recycling. In the middle, you can see there that the tank or the sink or whatever, bath or whatever you want to call it, is building up, stuffed with material, because the tap is running like it was, but the way of emptying it and turning it over and recycling that water through the sewage system is impaired. And the idea is to just attenuate, to just reduce very slightly the input by the tap so that it can cope with the slow drip and recycling out, and the material in the lysosomal space, which is toxic, goes away. So that's the principle. Next slide, please. It's a great principle. And Francis Platt in Oxford found a chemical that would block that reaction. This is the similar thing in a different part of the cell to the lysosome, very much like the statin drugs. She found a compound, it was an amino sugar. She found it about 1994, and we've been friends ever since. In fact, before that. And she said, what can we do with it for the sphingolipid diseases? And I said, how about Fabry disease or Gaucher disease? Because we knew that there were patients that might respond. Next slide, please. So we did the trial. We did a trial for this drug. It's a sugar. It's very related to something you find in various plants. And I was just a simple clinician who collected the patients and thought this would be a good target. And in fact, the drug worked. So by 2003, this oral therapy, a simple tablet taken 2 times a day, 3 times a day sometimes, was sufficient to reduce the formation of sphingolipids and it improved the simple external features of lysosomal disease. Unfortunately, it's still approved and used, and it's used for Niemann-Pick disease, type C, another lysosomal disease, which we thought and showed together might be a good target. But it works in mild to moderate disease, but is a second choice for people who don't respond to the other treatments for their livers and spleens and bones, and it has adverse effects. It causes painful neuropathy, diarrhea, and so on. And so it's not an easy drug to take, but it was approved. Next slide, please. And here it is. Here, people with a low hemoglobin in yellow there with Gaucher disease given this simple drug, and you can see that their hemoglobin improves. Next slide, please. Here is, sorry, I'm a bit slow holding you up. Here is the effect on the liver and spleen volumes. They get smaller. Very good. Next slide, please. So we began to feel, and here is an effect on the bone marrow, which is an abnormal signal on the left. Over time, this was done in the Netherlands by Carla Hollack, a colleague, and you can see the improvement in the signal going green or normal in the bone marrow signal of the spine, which is affected in the disease. Next slide, please. So here's eliglustat, the one that was approved, developed by Norman Radian's student, Jim Shaman, also a good friend, much more powerful, much higher affinity. Next slide, please. This oral therapy works for Gaucher disease. Huge clinical trial. It took about, 12 years. It was nearly 400 patients, followed up for up to 10 years in some circumstances. Next slide, please. And you can see that the blood counts get better and the liver and the spleen volume go away in these patients here. 22 started, 19 remained on the trial for more than 8 years. You can see an amazing effect. So this is a powerful drug, which was approved in 2004, 2014, 2015. Next slide, please. And the clinical trials show that it worked for people with big spleens or small spleens. It would normalize them in the period of exposure, which is tremendously good news. Next slide, please. And it makes the case, of course, for other things. Biomarkers, the key things of the disease itself. In fact, those sphingolipids in the blood all came down in parallel on treatment. Next slide, please. So it works like it said it works. But the last main frontier, of course, is the brain. And now I'm going to share some very recent information, and I might go on just a little bit too long, but not, I hope, too much. The brain, the last kind of frontier, because it's difficult to get drugs into the brain, even for gene therapy, as we know, but you can get small molecules or compounds, some of them, which will go safely into the brain. Next slide, please. And in Gaucher disease, of course, most of the people in the world outside the Americas and Europe have a very severe form affecting the brain, as shown in these two little children here on the left, and the young lady in more juvenile form who's now a mother and a graduate of British University. But they have been affected by the neurological manifestations. Next slide, please. I always hoped that if miglustat would work, this is what I wrote in that paper in the Lancet in 2000. We hoped that the introduction of this principle would in fact help the conditions such as Fabry disease, neurological Gaucher disease, and you can see it there. I actually wrote it down in 2000, GM2 gangliosidosis, what we're dealing with today with you. It could, a whole family of disease, a small molecule could get into the brain. Next slide, please. Well, the idea then that the therapy, apart from those ones on the right of the slide, would be substrate reduction therapy for Gaucher disease, can be applied, of course, to those in the same family, particularly where the brain is affected. Next slide, please. And eliglustat worked. Next slide, please. But what about another compound that would enter the brain that was developed by the company, Genzyme, then Sanofi, Genzyme Sanofi, now Sanofi Genzyme, that managed to generate venglustat, which is now an investigational product in late onset gangliosidosis, Tay-Sachs and Sandhoff, but also in patients with type 3 Gaucher disease. Next slide, please. Now I'll try and show you the evidence for this. Next slide again. All these conditions could be improved because the sphingolipids are common. So the trials, next slide, it's out of order, I apologize. Next one, please, sorry. Next one, next one, next one, next one, next one, next one. I apologize for this. Next one, next one, next one. Here we are. Let's go back. This is the compound. Can we go back one slide? This is the compound benglustat. It's easy to take. It enters the brain. It is excreted by the kidney, and it can be given once a day, and it's a very potent inhibitor of the same target. It is not approved in any condition or any country at the moment, but it is in clinical development, and this is quickly what I will share. I've done so before, but it's really looking good now, and it's encouraging. Next slide, please. So the idea is that it will traverse the blood-brain barrier and decrease glucocerebrosidase synthesis and ganglioside formation as well. It could work in Fabry disease. It could work in Gaucher disease that affects the brain, and it could work in GM2 gangliosidosis theoretically. Next slide, please. So here it is. We always hoped it would. Next slide, please. And so we took patients, and there's a trial at the moment called LEAP, which involves children and adults actually, with type 3 Gaucher disease. That is to say, the neurological form. They remained on their enzyme therapy for their systemic, their body manifestations, but for the brain, they were brought into this because they had neurological manifestations. Next slide, please. We have the results of a 1-year study, 52 weeks so far, in 9 patients. And you can see that the drug enters the brain. On the left panel there, it goes in. There was none before. And you can see that it goes into the cerebrospinal fluid, which is a measure of what's happening in the brain. Next one, please. So it actually enters the brain and the plasma after you take the tablet. And here, all the patients except one were exposed to the drug. And you can see that it reduces what we call GL1, which is glucosylceramide, the one I was telling you that come down. Yes, in the patients exposed to the drug, you can see that the concentrations of glucosylceramide GL1 and lyso-GL1, which is glucose, in the cerebrospinal fluid. So it engages, as they say, with its target. And you can see that the engagement is 30 to 60% in the CSF, which is what we'd hope for, what we'd expect. A primary endpoint at this stage of this stage 2 trial. Next slide, please. Now, this is quite difficult. I'm coming very quickly to the end. Next slide. So there was an improvement, it's safe, and there was an improvement in the coordination, ataxia, and also special studies of the brain by functional magnetic resonance imaging and the size of the brain by volumetric, measuring the volume of the brain very sensitively in the patients, studying what we call the activity of resting state networks. Next slide, please. This is very complicated to show, but the next slide will show it. Sorry. So here we are. So in the, sorry, can we go back one? Sorry, we overstepped. Yes. So this is the image which shows very quickly that there's enhanced patient who was not exposed to the drug on the right. You can see that blue mark in the circle there, up and down. That shows the resting state networks, parts of the brain, the hearing, the seeing, the speaking, the thinking, and the feeling those bits that are involved in execution of brain functions had improved connectivity in those taking the drug, but in those not exposed to the one not exposed to the drug, if anything, it got worse. Yellow is better, green is the same, blue is worse. So you can see more connectivity around the brain. This was in those patients, which is quite extraordinary. Next slide, please. What's more, the next one. What's more, in that those bits of the brain increased in size. There was a slight increase in the volume, but in the patient not taking the drug over the 1 year period, who deteriorated, in fact, you can see that her brain got smaller, and the ventricles that reflect the spaces in the brain got larger during that period, decreasing. So this is a remarkable effect, a significant increase in brain volume in the connected areas, in those responding, and with great improvement in their networks. And you can see in the other patient, an internal control by chance deteriorated. Next slide. These are very encouraging results. At this stage, it's only 1 year. There's not an approved drug. There's another year of this study. Next slide. And a whole program of work in type 3 Gaucher disease, which is quite a common disease worldwide and a very important area. We're just waiting for the next slide, nearly my end. Final slides. Just to say that based on this, these are the improvements that we see, functional and volumetric, indicate greater coherence in the brain, such that the entire brain is more connected. And these are the studies we have. We've started now in late onset GM2 gangliosidosis, patients above a certain age with Tay-Sachs and Sandhoff diseases, based on the probable lack of efficacy due to little or no residual enzyme in these children, no plan for the development in children below the age of 5 years, but a secondary population of patients, including those of juvenile GM2, were included in patients more than 5 years old. And that was expanded to 20 patients. This trial is now almost fully recruited in the adults who have a disease-controlled arm. That is, it's placebo-controlled trial on a 2-to-1 design. In other words, 2 years of exposure and measurement, a little less invasive than the one in type 3 Gaucher disease, but a very intensive trial with a control arm in the adults. But the likelihood, 2-to-1, is that patients will be on the active drug. We don't know whether they're on the active drug or not, but we follow the patients very carefully and a PI on this trial. And then the secondary trial is for children, younger children who've been on it on an open scale. A very small number of those have been able to join. And that's where we hope we're going to learn. We're now 1 year in, approximately, to those that have been recruited. And I'm sure in another year's time, or even later, even earlier than that, we may have some indications of how patients are going to do. Because, of course, the GM2 ganglioside has exactly the same relationship to glucosylceramide and venglustat as it does in the other diseases, where there's success and where we see it operating. So, and I'm sorry to go on too long time, but thank you very much indeed.
That's great. Thank you, Tim. As always, a really interesting topic that we've heard and seen such advancement on in the last, I would say, few years, especially when we first started talking with GM2. Obviously, you were instrumental in us establishing the CAT Foundation back in 2011. I remember us having a discussion in Cambridge at the time about all these potential therapies. So it's really, really exciting. So,
yes, takes so long, but yes,
we're getting there.
10 years though. Yeah, we're getting there very much. Very much.
So,
yeah,
Tim will be joining our panel later today. So we'll say goodbye to Tim, and I'm going to welcome in our next speaker. So it's a great pleasure to welcome in Taylor Fields from Intrabio. Taylor is going to be talking about drug repurposing for GM2 and about the study that they've been running with Intrabio. Now, I've known Taylor since I think about 2017 when we first met, with Mary Factor from Intrabio as well. And it's really amazing to see the speed at how they've been able to get this study up and running. And it's really exciting, the results they've seen. And I'm really kind of honoured as an organisation, not just us at CATS, but also the other consortium members, that we've been actively involved in this study. And it's been very patient-centric and taken on board our feedback. And I think that's really important. So I'm going to hand over to you, Taylor. I'm going to add your screen. And then, yeah, there you go. Thank you.
Thanks so much, Shane. Can you, and thank you for that lovely introduction. Can you see my slide deck okay?
Yes, we can.
Great. All right. Well, lovely to be presenting today, and thank you so much for this opportunity. I'm going to give an update on Intrabio's development of N-acetylleucine for the treatment of GM2 gangliosidosis. So before I go to our most recent exciting study results, I want to just give a quick overview for anyone who may be new or less familiar with our development program about our lead drug compound. So we are developing a drug called N-acetylleucine, which is a chemically modified version of the amino acid leucine. Now, this chemical modification, it can come as 3 forms, and this is important just to clarify the actual drug we're developing. So for modified amino acids, you can have stereoisomers, or you can have a racemic mixture. A good way to understand this is stereoisomers, a left and a right, are like your left and right hands. They look very similar. They essentially are the same size, same shape, but they do different fundamental things. And this is the same thing as the stereoisomers in N-acetylleucine. So you have the L and the D. And again, they're mirror images, but they're flipped, and they do fundamentally different things in the body. At the same time, you can have a 50-50 mixture, what's called a racemic mixture. So if you imagine you cross your hands, you've now got both the L and D form in, but again, this is going to do a different thing in the body. So those 3 compounds, again, although they're all versions of N-acetylleucine, they are fundamentally different drugs, and they work differently. They have different pharmacology, different pharmacokinetics. Our initial development began with that racemic mixture, so the 50-50 mix, which had been approved in France in the 1950s for the treatment of acute vertigo and goes by the trade name Tanganel. Tanganel has never been approved anywhere else in the world. It's never been approved for any other indication other than acute vertigo. And there's actually very limited data on its efficacy for acute vertigo, but the 1950s were a different time for drug development. The one thing we do know about the drug is that it has a very benign safety profile. It's very well tolerated, and it's orally administered. So back in the 2000s, one of our founding scientists, Professor Michael Strupp and Dr. Tatiana Bramova, had the idea that based on what they knew about the drug at the time, it may work for some patients with cerebellar ataxia. And they began a compassionate use program at the University of Munich, where they very quickly saw symptomatic and cognitive improvements in their patients. And they then spread those compassionate use programs to other disorders where there's involvement of the cerebellum, including GM2 gangliosidosis. So over the past 10 years, we've had these observational programs where we've now seen both the symptomatic and the long-term neuroprotective effects of the drug. Those case series are all published. They're available via our website, and you can also email me if you're interested in reading those. In parallel, our wonderful team at the University of Oxford, led by our scientific founder, Fran Platt, started to investigate of those 3 compounds. So I'll just go back quickly, the L, the D, or the racemic mixture, what the most effective form was. And we ran a lot of studies in different animal models and cellular models, and we identified that the individual L enantiomer, N-acetyl-L-leucine, is the active enantiomer that is responsible for the drug's activity, but also has a superior safety profile when administered independently. I'll get to it in a moment, but we now know that the D inhibits the activity L, and again, gives us reason to believe that the L administered by itself works better and is safer than either the racemic mixture Tanganel or the D by itself. So we are developing that individual L enantiomer, which we call IB1001. It is not currently approved anywhere in the world for any indication. And we've made a few additional modifications. So we know that patients in the GM2 community, for example, have swallowing issues. So we formulated the drug as a sachet that can be mixed with water. And I'm pleased to now add with juices and milk. And is compatible with PEG tubes and feeding tubes. The drug is transported across the blood-brain barrier. I'll touch on that in a moment, but it's also distributed to major organ systems. We know it's very well tolerated, has an equally benign safety profile. And again, we're investigating this for both the short-term symptomatic benefit, but also the long-term neuroprotective disease-modifying effects. So to spend a minute just touching on the mechanism, we've recently discovered that a question we get quite often is, does the supplement L-leucine do the same thing as N-acetyl L-leucine? And the very short answer is no. L-leucine can be available as a food supplement, but even if you took hundreds of kilograms a day, you would not receive the pharmacological benefit. And this is because with the N-acetyl group, that chemical modification that's added on, we now know that when that happens, it allows the compound to be picked up by a different transporter in the body. Transporters are mechanisms by which drugs and anything you eat really is distributed. And we know that when the N-acetyl group is added on, monocarboxyl transporter picks it up rather than the standard amino acid transporter. And this allows the drug to be distributed throughout the body, again, to the brain, to major organ systems in much higher quantities. The metabolism is very simple. It essentially then cleaves off the N-acetyl group and it delivers L-leucine to the cells in a revolving door transport mechanism. So we see that this MCT transporter is able to identify cells where there's a high buildup of lactate, so obviously some sort of distress, and it not only drops off the L-leucine, but it picks up the lactate, which we think contributes to this disease-modifying effect. The publication of this is all there. That's my simplified version, but happy to answer any questions on that. And then once L-leucine is dropped off in the cells, we see it does a number of things. So for this drug, we know that there is not a single mechanism. It's got polypharmacological effects. It's doing multiple things. We do not know the primary mechanism, but our best guess at the moment is that it relates to energy metabolism. So Professor Platt and her team have demonstrated that the drug normalizes energy metabolism and it kickstarts ATP production. So essentially the cell is making more energy, which is allowing it to function better, allowing other processes in the cell to function better. And it both can restore the function of a damaged neuron, but also protect the function of a neuron. This isn't on the slide here, but we see as a secondary effect, there is a normalization of lysosomal volume, of lipid levels, of cholesterol. So we see a substrate reduction effect, but in other studies, what we've done, we see the drug is highly synergistic with miglustat, which leads us to believe this substrate reduction effect is a secondary effect, but not the main reason the drug is working. In the literature, we know that the drug normalizes neuronal membrane potential, which relates to cellular signaling and is part of the reason we believe it has this effect on cerebellar symptoms. The data you see at the bottom here is actually with the Niemann-Pick disease type C mouse, but we've done the same and replicated the same data on the GM2 mouse model. I'll start with the panel on the bottom right. What you see here is a functional assessment. So we have a healthy MPC mouse, an untreated non-healthy MPC mouse is the second line there. And then we have the MPC mouse that's treated at 8 weeks. So when the mouse is already ataxic, it already has symptoms, and this mouse has only been treated for 1 week. So that short-term treatment mirrors what we do in the clinic when we look at patients, say, for 6 weeks. And we see even when you introduce this drug acutely for a very short period of time, there's a very significant symptomatic improvement in functioning and gait and ataxia. And then the final line there, the 3-week start, this is when the mouse is treated before they're symptomatic. And we see essentially a normalization of symptoms. So it's very close to the healthy mouse. And like any drug, the earlier intervention, the better the results. So we're not unique in that regard, but we do see the earlier start, the better the patient functions. The panel on the left bottom is then the results that we see with survival and lifespan. And what we're seeing in the animal models and what we've now seen in the clinic is about a 10% slowing of disease progression and extension of lifespan. Interestingly, when this is used with miglustat, there's almost a doubling of that. So we're hopeful that substrate reduction therapy in GM2 will also be available and will have that synergistic effect. On this slide here is some of the work from Fran's lab just in the GM2 mouse model. And you can see here the untreated versus the treated mouse. And it's nice to just illustrate what those lines in the previous graph mean in regards to functional improvement. So obviously the untreated mouse is unable to complete the test, has a deteriorated motor function and limited high limb mobility, while the treated mouse is able to complete the test. And again, not pictured, but there's a significant increase in lifespan. So it's a very rapid overview of the data that we have. We have, as I mentioned, published everything from our in vitro studies to the in vivo studies and the observational studies. But all of this supportive evidence was the basis for the multinational clinical trial, which we've conducted for GM2. And this study we ran to investigate both the symptomatic, but also the long-term effects of the drug. So in the parent study, we looked at patients during a baseline period, a 6-week treatment period, and a 6-week washout period. We know that when the drug is introduced, we believe symptoms improved, and that when the drug stopped, there's a return to baseline. So that washout period really served for us to see not only are patients improving on treatment, but is there a deterioration when they stop the drug. We've got an extension phase ongoing now to investigate the long-term neuroprotective effects. And one of the reasons I want to highlight that we do this symptomatic study is because with our drug, we're in a very unique position that we can demonstrate benefit rather quickly. And so from a regulatory perspective and for a patient perspective, we've always sought to demonstrate that symptomatic benefit first and foremost, because it means we can get the trials done quicker and the drug approved quicker for patients. It does not mean that we do not believe there is a substantial long-term disease modifying effect, but that we're in a position to run shorter trials, which lead to shorter timelines for the drug to be approved and accessible to patients. So I just wanted to make sure I made that point. As I mentioned, we've now completed the parent study from that, and we were extremely pleased to announce that the trial was very positive. It met both its primary and secondary endpoints, and the clinical relevance was we saw patients walking with aid to unaided, a significant reduction of ataxia and tremor, and improvement in fine motor skills, very significant improvement in speech and swallowing, all functional changes that are hugely relevant to quality of life. There were no serious adverse reactions that were related to treatment. The drug was, again, very well tolerated. And as I'll get to in a moment, we saw that the drug significantly improved symptoms during treatment, and then there was a deterioration during washout. So the results you see here are primary and secondary endpoints. The boxes in blue represent the changes during the treatment period. The boxes in orangish yellow represent the changes during the washout period. And again, we're seeing that nice bell curve improvement during treatment, deterioration during washout. We also have data which is even further encouraging from another lysosomal storage disorder that's related to GM2, Niemann-Pick disease type C, where we saw essentially parallel results. We saw a significant improvement during the treatment period. The deterioration returned to baseline during the washout period, and the drug was very well tolerated. In both of these studies, there was a very broad age range and disease severity studied. So in the NPC study, it was 7 to 64. In the GM2 study, it was 6 to 55 years old. And we really had patients of every severity, from juvenile onset to late onset patients, full range of SARS scores. So it gives us very, very supportive evidence to develop the drug for all ages and all patients, which is what we're continuing to do. So just to summarize the totality of the evidence that we have thus far for GM2, we have obviously the successful multinational clinical trial that demonstrated very positive, statistically significant results. We have the supportive trial data from the NPC study and the earlier observational studies in GM2, where again, we're seeing this rapid symptomatic benefit, but also a slowing of disease progression. We've got in vitro and in vivo, so cellular and animal studies and GM2 models that directly correlate to the effects we see in the clinic. And we've now really elucidated the mechanism to help explain why it's working for GM2. We have only ever seen that the drug is very well tolerated, and the data from our most recent trials support that. And we have evidence now that IB-1001 has a very favorable risk-benefit profile for GM2 patients. So as our next steps, we're currently meeting with regulators in both the US and Europe to discuss what's necessary for GM2. For NPC, we know in the US that a placebo-controlled trial is required. We are trying to determine if that's necessary for GM2, if data from the placebo trial for NPC is supportive, and everything we can do to get the drug to patients as quickly as possible. So on this last slide here, I've just listed my contact details if you have any questions, the link to the clinicaltrials.gov website where you can see details of the phase 2 trial. And then the trial results for the GM2 trial have been uploaded to a preprint server so that it's available for the entire community to see. So those are the details there if you're interested. That is my presentation. So I'm happy to take any questions. Thank you.
Thank you very much, Taylor.
Cheers, how are
you? Nice to hear you. I also know the work you have done since 2017 with trials, with opportunities for the patients. It's too great and too gratific, and I think for you to satisfy and to see it on paper. So very congratulations to all the team at IntraBio.
Thank you. Thank you very much.
We have a question.
Sorry.
We have a question from Katia Lopez. I guess this is sent to you because of the moment. I would like to know if can you answer this question. Are there any plans for trial in infantile form of Tay-Sachs?
Yeah, so our discussions with the regulators have been very positive in the sense that because our goal has been to demonstrate the symptomatic benefit, the reason that our trials haven't included younger patients is unfortunately infantile or very young juvenile patients can't perform the tests that are done in the trial. So they can't do the 8-meter walk test. They can't do speech tests or the 9-hole peg test. That said, we do have data from observational studies in patients as young as 6 months old. We know that the drug safety profile for all ages is very benign. And the regulators have been very, very welcoming to the idea that the drug will be approved for all ages, given the safety is demonstrated. So our hope at the moment is that we do not need to run clinical trial on infantile patients, but that we can go for authorization for all ages on the basis of the data that we have now.
That's fantastic news. Great. That's great. So thank you very much, Taylor, for being here today with us, for sharing all this work you have done. And we are going now to introduce the next sessions. Hello, Ruben. Welcome to the Tay-Sachs and Sandhoff European Conference. Dr. Ruben Giorgino has more than 20 years of experience in the pharmaceutical industry. He has a vast experience in the lysosomal storage disease area, including clinical development for neuropathic disease. He's now head of clinical research and development at Asafaro, developing a patient-centric natural study in GN1 and GN2 gangliosidosis. So it will be very nice to hear you, Ruben. Thank you for joining us.
Thank you very much, Patricia. I hope that you can see my screen at the moment.
Yes, okay. Great.
I will put it in full screen. Is it visible in full screen now?
Yes, perfectly.
So, thank you very much. Thanks a lot for giving Azafaros the opportunity to join this conference. Really, and to me, it's an enormous pleasure to speak in the same session as other colleagues in the industry, but also, and most importantly, with Professor Cox, whom I have the pleasure to meet him now 16 years ago when I was working on Nilstat. So thank you very much for this opportunity. Azafaros is a new company and is a Dutch company which was founded in 2018 based on a series of patents from the University of Leiden, and in particular from all the experience that Professor Hans Ars, who is one of those like King Cox and Frank Platt, who have really set the stage of research in the area of lysosomal storage disorders and glycosphingolipidosis in particular. So the company was founded to focus on rare genetic metabolic disorders, and in particular CNS, of course, disorders. Before going to the topic of today, a short introduction on what we are working now. The name of, we don't still have an official name, we still have a code, which is AZ3102. But as I'm going to share in a second, we are at clinical stage since some months now. And it's a small molecule. less than 500 Dalton. And as such, we have seen high penetrance into the brain, and importantly also prolonged and steady levels into the brain. The mechanism of action of SZ3-102 is dual. So one is the, let me say, classic SRT effect, substrate reduction therapy effect that Professor Cox has been extensively talked about. So to reduce the synthesis of glucosylceramide as first committed step for the synthesis of gangliosides. And there is lots of science around it. The other point, and that was also the effort of Ansar's group, was also to find a molecule that could inhibit at very low concentration, so very potent and selective inhibition of another enzyme, which is the neutral, non-lysosomal glucosylceramidase. And in the future, we're going to tell more about that, but this is also to normalize the pH of the lysosome, and importantly to avoid that metabolites or catabolites of glucosylceramide, such as sphingosine or of ceramide, do increase within the cell. So a mechanism of control. I was telling you about the potency, and which is very high. I mean, for both enzymes at nanomolar concentration, at single digit. There is no effect on the lysosomal glucosylceramidase, which is very important because we need to save the function of the lysosomal glucosylceramidase. But from a tolerability perspective, it's very important that the product does not inhibit what the enzymes which are called disaccharidases. So those small bowel digestive enzymes which are breaking down complex sugars into simple sugars. And this is important because this is an hypothesis for say believing and observing, because we have not observed so far gastrointestinal side effects, both in animals, but for the limited experience that we have now in healthy subject, also in human. It has a kinetics which would allow a once daily administration, and we're also working to develop a pediatric formulation, meaning something that young children could swallow more easily. Now, the topic of the presentation today is to talk about the Pronto study and all the infrastructure that we have put in place for this study, which is a observational, so there is no drug administration involved into this study, but we want to characterize as much as possible the trajectory of neurological disease in children who had an onset of either Gen 1 or Gen 2, the late infantile onset forms and the juvenile onset forms. A question that we hear very often, and actually I've heard very often recently, is but do we really need another natural history study? No, isn't the evidence that we have at the moment sufficient? Why do we need to get more information on that? Well, my point is that we need more information. I mean, so all the information that we have so far comes from the literature. They come from small sample size, maybe from a single study, and from and also we have sparse information. So we see the need of quantitatively characterize the progression of the disease with a protocol which is the same across the different centers, where the assessment are done all at the same time. So this is really important. We strongly believe that this will serve to all the community, not only to us in case for future studies with investigational drugs. So we focus on neurological manifestations. We dream, let me say so, to have the possibility to build a disease progression model so that we could identify factors behind those who are well known, such as age at onset or genotype, that could predict the progression of the disease. This is a picture of things how they're going at the moment. We have 2 sites activated already, in one in France, in Paris, another one in Germany, in Munich. We have now 18 sites selected in 6 major countries, as you will see. So things have started moving finally, and we at Azafaros, the clinical team, is very happy to share this news with you. So, and we go back to the point of the patient-centric approach. I have to say that honestly, I wouldn't put even under discussion that a study, any study, must be patient-centric, because the patient, of course, is the key entity within any study. So all companies or industries should avoid to make studies which are purely academic or scientific exercises. So, of course, one of the main objectives from our side is, and also coming from the recent experience with the COVID, to make the study as much easy as possible. So to avoid stress to the children, avoid stress to the families, and from a methodological point of view, to avoid too many missing values. So we worked to make the study as much decentralized as possible. Maybe we are not yet in a situation where the study is completely decentralized, because patients will have to go to the investigational site, not too often, every year, but we are trying to decentralize it as much as possible. So we have all the telemedicine virtual visit platform, which has been used in other studies and is well validated also in terms of security and prices. We have developed some patient-reported outcome questionnaires that can be accessed through the telephone or through the computer. And moreover, we have also initiated a project with an artificial intelligence company in the US, PhysIQ, for an electronic sensor that must be worn on the trunk, that will collect and transfer to a server a billion of information to characterize vital sign on one side, but also information related to gait, to balance, and other characteristics of all the subjects that were going to be enrolled into the study. Inclusion criteria. The age range goes from 2 to 20 years, chronological age at study start. Of course, with a genetic confirmation of either Gen 1 or Gen 2 gangliosidosis, Tay-Sachs and Sandhoff. Of course, it's very difficult to, in this context, to define appropriately, how the forms that we want to reach. But in a way, at the moment, we would like to focus on what is normally called the late infantile juvenile form. So the definition would be to have subjects who have reached a normally development milestones at the first birthday, and then in case started to have onset of the disease after the birthday, which means could be at any time between 2 to 20 years. With a minimum level of impairment of gait and speech at baseline. And to do that, we are using a scale, which is called the SARA scale, which stands for a scale for assessment and rating of ataxia, and I'm going to spend a second on that. Very frequent question that we get from families, from patients group also is, if my son or my daughter we want her to be enrolled in another study, is this possible? And the answer is, of course, yes. We promote that subjects could enter an interventional study with a therapeutic. So at any point of time, patients will be free, let me say so, to join an intervention study. So for a natural history analysis perspective, of course, we will not include into the analysis period of treatment with investigational drugs or with any drug that, according to the investigator, may interfere with the appropriate assessment of disease progression. But of course, we want to assure families that they are going to be free at any time to join any other study, of course. So, as I said, the primary outcome measure will be based on this SARA scale, and we're going to look fundamentally as primary outcome measure at changes of two important components of the ataxia syndrome, which are gait, and the SARA scale has a grade which goes from 0 to 8, and the speech, mostly speech articulation, so dysarthria and there was case anarthria, with always the SARA scale. This is a little bit more about the SARA scale, which was developed now in 2006, or not now, 2006 from the University of Bonn. It has been validated for several performance aspects. And in particular, we are going to use, as I was mentioning before, a remote assessment, which has been developed always by the same university, by the DZNE group, the University of Bonn. And we're going to train, the investigators for an appropriate assessment. And again, this is going to be the key component of the study. These are pictures of the beautiful daughter of my colleague, Cécile Luzy. You may have seen her name in the references of Professor Cox. Cécile has been certainly the most important person at bringing miglustat to approval in Niemann-Pick type C disease now 13 years ago, and we are working back together. What we are going to assess fundamentally is the gait, as you see, the stance, the sitting, and then some dysmetria-related tests like the finger chase test, the finger nose test, the heel shin, and also the fast alternating hand movements. The Greek name, diastodocokinesia, is too complicated for me to mention. But these are all part of standard neurological assessment. We are not inventing anything here for the purpose of this study. This is standard neurological assessment for for cerebellar ataxia. Of course, we are going to look at other outcome measure, the other component of the SARA scale, also the motor function measure, which is I'm going to spend a second in a while, is a quite commonly used scale in neuromuscular disease, for instance, in SMA. A very basic performance test, which is the timed up and go, and then a Likert scale for swallowing. But let me tell you a little bit more in a second. So the 32-item MFM is, again, pretty common. It doesn't take, this will be done at the site. So it's not going to be done in telemedicine. It's going to be done on a yearly basis by trained physiotherapists. And there are 32 tasks which are assessing different motor function, from standing to transfer, the axial and proximal motor function, as well as the distal motor function. So, and also in this case, we are putting in place a training session for all the personnel involved into the study. The time up and go is pretty easy. It's measuring the time that the subject is standing up, walking with for 3 meters, turning back another 3 meters, standing up and sitting down. So there are 7 different steps which are measured in terms of time. This is used very commonly if the subject is not able to perform the test. Can be supported by a family member or by the personnel. But this is another important point that we want to consider looking at the progression of the disease. And then the important part is truly the patient-reported, or in this case, the patient-reported outcome. So we are going to ask parents to fill up through the telephone a questionnaire, which is quite common in this, to evaluate cognitive impairment and also developmental milestone in children, which is called Vineland Adaptive Behavioral Scale. I'm going to spend a second on that. And then the BSVC, again, which is more of the burden of the caregiver. So to further understand the burden of the disease management from a caregiver perspective, and then a diary of the disease. And again, I was mentioning before this patch that is commercially available and approved by the FDA for vital signs measurement, but we have already algorithm to assess a series of gait and balance parameters. So this is the Vineland. Maybe some of you are familiar with that. It's assessing different domains from communication to daily living scale, socialization, motor skill, and maladaptive behavior. We're going to ask the parents to compile it every six months. It's taking 45 minutes, which is a long time, but we will allow, and the study will allow parents to compile the questionnaire, not everything at the same time, so they will have more than 1 day to compile on their telephone or on the computer the questionnaire. And of course, this is an important part from a scientific perspective, because we were going to accrue data that will support the clinical relevance of what we are going to see and measure through the SARA scale. This is the Bardet scale for family caregivers. Again, it's not specific for gangliosidosis, but this has been used in many other studies looking at the impact, the psycho-emotional impact that the caregiver have managing their relative, in this case, their children with the disease. So this is an example of the e-diary. So we're going to collect information on seizures, on choking episode, on respiratory tract infection, and this is going to be compiled by the caregiver. So again, I would say family focus, at least, if not patient directly focus. And again, this is what I was mentioning before, this patch. It sends through a telephone. We will have a dedicated smartphone to do that, sending information to a server and applying different algorithms of artificial intelligence. We are going to measure, as I was saying, several aspects related to gait, walking, also symmetry of movement of the trunk, and etc. So I hope that this will provide also further insight into the characteristics of the progression of the disease. Before leaving the call, let me take the opportunity to present 2 slides on AZ3102. As I was mentioning before, the product is at clinical stage since the beginning of this year. We have completed, I would say, successfully in September the first human clinical studies in healthy subjects, administering a single dose of the product and multiple doses over 14 days. And finally, 10 days ago, we have seen the results after unblinding of the data. And we're really very happy, enthusiastic that there were no signs of safety issues, an excellent tolerability profile because there were no GI issues, which is quite important, no neurological adverse event. In this case, suddenly, although there was no real rationale, but we did look at potential abnormalities in the cardiac rhythm, and there were no signals. A very good pharmacokinetics, so the half-life is compatible with once-a-day administration. We assume, based on the data, that, or we infer that the bioavailability is more than 90%, which is good, and there is no accumulation, substantial accumulation over time. But last and not least, what we have seen is a large reduction of glucosylceramide level in the blood, which, and on the other side, an increase in this, in the cerebrospinal fluid. And this is important for us because it's consistent with what we wanted the product to do, I would say, the dual mechanism of action. And really, the size of the effect is very, very large. And we are happy for that. Last slide. but I assume that you would like to know more from us, from Azafaros, what are we going to do in parallel with the Pronto study or after the conclusion of the phase 1 study. Well, normally what companies do is to go and discuss their plan with health authorities to challenge the plan, because sometimes we tend to be very optimistic. But what we're going to do is to discuss our plan with the FDA and possibly also Europe. And as soon as possible, we would like to start a study, a long-term safety and efficacy study with the product in children with late infantile and juvenile onset of the 2 diseases. We have decided to design the study in a way that we are going to have primary objectives which are reflecting clinically meaningful measure. So we don't go for surrogate endpoints. There was already discussion today about the placebo arm. We would like to convince the authorities that a placebo arm is unethical, unfeasible, and so also the data coming from the non-natural history study will serve as a control for the study, for the interventional study. And more importantly, I think also for the audience, is that we want to go to many more countries. We acknowledge that we need to be as much global as possible, so new countries and also more sites for the intervention study. There is commitment from our side. I speak on behalf of the company that we will inform the patients, families, community on the next step as soon as possible and upon the definition of the plan. This is my last slide. Again, thank you so much. Thanks a lot. It has been really a pleasure to be at this session today.
That's great. Thanks, Ruben. It's been really exciting following the work you guys have been doing. I think the first time we met was in the World Conference back in 2020, which wasn't actually that long ago, but it seems like a long time, kind of pre-COVID days. But to see how far you guys have been able to move things forward, it is really exciting for our community. And really, we can't wait to see what happens in the next couple of years. So yeah, thank you again for joining us. I'm sure you'll be joining on the panel later today. So if anyone has any questions, please prepare those and we can chat them. So thank you.
Thanks
so
a lot. All of you. Thanks.
I'm now very excited to welcome in a good friend of mine, an ex-colleague, Liam Eaves. We first started working together a few years ago. Liam's going to give us a really interesting talk about Sanogenetics and their approach to recruitment and the technology that they've been using. So Liam, I'm going to hand it over to you just to maybe introduce yourself in a bit more detail and to run through your presentation. So there you go.
Fantastic. Thanks, Dan. Thanks all for joining this session. I thought what would be useful is these slides aren't pulling up right. Dan, can you see those slides? They've gone a very funny pink.
They've gone a bit pale. Let's check the next one.
Okay, yeah, that's on. Well, okay, so I'll talk to them. I've got my other slides up. So just a bit of an overview in terms of where I'm going with this presentation. Wanted to give you a quick introduction to myself, then a high-level view of precision medicine, and then start to dive into some of the first principle problems in research. And then from there, I can then start to explain around how Sano is doing things and how we're trying to rethink some of these issues. So as you, I think these slides have gone a bit design funny, but essentially, as you'll all know, that precision medicines are really on track to surpass a lot of non-personalized medicines in the next 5 years. And essentially that this is all around tailoring treatments that are using data. And we believe that there's obviously a revolution that's happening. So from our perspective, this is a huge opportunity, but the industry really needs to adapt its thinking and also its philosophy. Just before I dive into some of the problems now, so one of the I've been very fortunate that I've been able to run clinical trials for the last 15 years. I have run every aspect of a clinical trial and every function right across from observational to phase 3. And at Sanogenetics, I head up the the enrollment solutions. And that's what we're now going to talk into a little bit. So there's, as I mentioned before, that there are huge problems when it comes to patient recruitment in precision medicine, but also any other types of clinical trials as well. We all know that 80% of clinical trials are delayed. It's a huge cost to pharma. Also, obviously, the biggest impact is to the patients themselves. And we know that 90% of these drugs fail in trials. Yet with genetic data, this increases the success rate significantly. So what I wanted to look into is really some of the first principles, problems that span across all types of research. So when we think about research, it's a very siloed approach, right? We have our R&D function that are interested in gathering information, data, and so on and so forth. Then we stop that. We then move into the observational, then phase 1, phase 2, and phase 3. So the methodology that we start to think about when recruiting patients is very stop and start. We start an activity, we stop it, and then there's this huge amount of time between these different phases where there's a lot of lost opportunity. So that's one of the first principles. The second is that that patients that are actually enrolled in these studies are not necessarily engaged with ahead of the clinical trials. So as I mentioned before, is the whole philosophy or the whole principle is that we start from scratch, one after the other. So we lose a lot of interest from individuals who may sign up for one, but may be suitable for another study later down the lines. The third problem is very much around the fact that it's very difficult to recontact with these individuals. So we find that groups like biobanks that have genetically profiled individuals, they can't then be easily recontacted for clinical trials. So that the genetic data has to be regenerated again at a high cost further down the process, which obviously results in a huge amount of screen failures and cost and a massive impact to time. Then there's a lack of patient give back. This is one that's very dear to our hearts, that we found that individuals, patients are not necessarily kept up to date with what's happening in the program, and also that their data is not given back to them so that they can understand some insights for their own personal self. Okay, so this slide isn't working again. So we're trying to rethink we need to rethink a lot of these recruitment problems. So as I mentioned before, only 80% of trials fail. Yet, why do we start late, knowing that there's a high % probability that a lot of these clinical trials are not going to deliver on time? So we should be considering how do we start earlier and also fill those gaps in the stop of the clinical development process. That an understanding of why can't patients be recontacted and leverage the past data, whether that's genetic or medical, so we can significantly increase speed. One of the big insights that we found when we were working up solutions was that when individuals actually have access to information, particularly when it's genetic information from what we've found at least, is that they're far more highly engaged to research than those that are not, to a factor of 4. And that we also must be working as a collective rather than working in isolation. We find that most clinical trials typically only take a few methodologies to actually find patients, such as going to site, social media, and they're usually the big drivers. But where we think that actually there needs to be more of a network effect in which that everyone benefits within the network. This slide design have gone to so essentially what's happened now is that in terms of Sano, our story was that as scientists we were really frustrated by how expensive and time consuming it was to recruit patients with specific genetic profiles to take part in research, and also then to get access to their high quality data sets. And then from the other side, as patients, there was this we knew that there was a huge interest in advancing research, but it didn't seem like the patient experience was really thought about. So issues around things like transparency and engagement from communication and updating. So we decided to look at some of these like key problems and start to build a solution that was going to help recruitment across all studies. So this is our sort of schematic of looking at actually how and what we do to support the process. So the first thing to notice is that the platform, it has many functions to it, which I'll go into in a minute. But what we try to do is we try to cover the whole breadth of the clinical development pipeline, so that we're in a position in which the individuals or patients can be engaged very early, and they have a relationship with us throughout a long period of time, as opposed to the very transactional aspects of research, which, as I mentioned before, is very stop and start. Obviously, it's not the case in some disease areas, but typically what will obviously happen is that groups will set up these microsites as a way of engaging with patients and letting them know what's happening within their therapy, as opposed to looking at how do you build those individuals up from across a long period of time and keep them engaged, keep them warm, keep them interested and updated with the information. So Sandhoff's created a platform essentially to do that. So it's about, and the platform consists of a number of modules. So it's a recruitment. So we look at recruitment as a network. So we actually do some of the traditional stuff like online, marketing and things like that. But it's much more important for us to actually work with patient groups to essentially help them understand some of the clinical trials that are maybe suitable for their group. And also we support patient groups by actually providing them technology that they can run some of their their websites and things like that. So it essentially streamlines information flow for patients, patient groups, sponsors, and ourselves. So in this, in the portal as such, that patients are able to not only find out information about particular clinical trials that may be suitable to them from both a medical but also genetic perspective, they're also able to consent and conduct pre-screening activities throughout the process, which basically means that they're able to come onto the site, find out more about the study, and then also take part in screening for that particular study to see if they're interested. Now, if there's a genetic component to that particular study, one of the big things that we're able to offer is actual genetic test that can be sent to for at-home testing. So as opposed to, I know there's a big drive for decentralized trials at the minute, and that plays obviously nicely into that. So individuals essentially receive a kit. It's a saliva-based test in which that they send off the sample to us. That's processed. Individuals are kept updated with that information and where it is, their process, how long it's going to take to process, and so on and so forth. So they're engaged in the process throughout. The other aspect is we can plug in and actually pull out medical records. So we can do medical record retrieval for around 7 years on average within the US, and we're coming soon to a number of other countries as well. And then the big part for us is, as I mentioned, is around the engagement. So I mentioned before around individuals with personalized genetic information or reports. They're much more interested in research. So what we've actually built is part of that give back from us, and the transparency is that patients, as they come through our process, they own their rights to their data, which I think is should be the case in all studies. what we actually do is that because we then have their genetic profile, we're then able to give them personalized genetic reports that relate to them. And what this does is that, going back to the slide that I mentioned before, is this then engages individuals so that there's interest in, and they get to find out a little bit more about themselves, as well as, obviously, as I mentioned before, about the research, which means that they stay on this platform and are engaged with it in a continuous fashion, rather than just being sort of left and said, okay, yeah, the trial's done, we're not interested in you anymore. We can actually keep individuals engaged throughout and help them learn a little bit more about themselves in the process. We also have the ability to generate information, questionnaires, and so on and so forth from individuals. So so far that Sano's supported 20 plus personalized medicine programs within rare disease and also chronic conditions. As I mentioned before, is that we're very heavy, or we lean into that patient engagement. That's really important. And the stat at the top that you can see there, around 80% of participants would be disappointed if we didn't exist, is for that very reason, because we're constantly trying to give back. It's also accessible to, it doesn't matter, we've used, it on the elderly population, visually impaired population throughout. We're currently in situ in the US and UK and Australia, and we're also expanding into Europe early next year. So going forward, our vision is really to enable this personalized medicine, or enable personalized medicine developers to digitally access 1 billion or more people with rich omics and medical data. And essentially what we're trying to fill is this middle bit. You can't see the diagram because the design's gone a little bit too cock, is that these individuals come through the Sano platform, in which that we're then pulling the relevant information from either from existing databases, or we're actually adding on additional information to that individual so that we can hopefully help develop these medicines quicker for patients. So that's everything for me. Thank you. I think you've got a bit of time back, Dan.
Thank you very much, Liam. Very interested and quite impressive to hear you and remember what we were talking yesterday about the importance of the data collection, how we need to access to more data. After listening to our previous speaker, Dr. Ruben Georgino, explaining how they want to carry on their study globally, in the most globally possible way, through virtually access. And also, of course, we live in a very global world, so this change in the medicine will also mean that more people can access, which is a problem in diseases like Tay-Sachs and Sandhoff disease with a very low prevalence. So it's amazing to hear you. Thank you very much for your work, and thank you for joining us today.
Thank you.
So now we are continuing with our conference with Dr. Eugen Menger. Hello, Doctor. Welcome to our conference. Dr. Eugen is a principal investigator, founder, and CEO of the SpinCS nonprofit organization in Germany. He has dedicated himself to research in the field of lysosomal storage diseases and has already significantly contributed to the advancement in this field of study. Thank you very much for joining us today, and you can start now. Thank you.
Thank you very much, Beatrice, for this kind introduction.
Dan is loading your files, so
yes.
We will be ready in 1 minute. If you want to start introducing yourself and the work that has brought you to this conference today.
you have already said that I 2 years ago have founded the Sphinx, and it's an independent institute, and it gives me the chance to make clinical research what was in 25 years not possible in a university, and that's a challenge, of course, to have all the organization in the hand and to be responsible for everything. But it's also the independency and make such a trial, which I like to report in a few minutes, like 18.1, and it was an idea long ago to make in gangliosidosis a natural history study, but to be true, it does not work on the university. But now it's working, and now are the slides there. And my talk is about multi-disease national natural history study in Germany. And next slide. And the name of the study is 8-in-1 stroke. And what I'm presenting today is quantitative longitudinal natural history of AT gangliosidosis. And what I like to present is the conceptual framework of the study and baseline data from the study in Germany. Next slide. The idea and the concept was not Sphinx alone, not me alone. It was a cooperation, hand in hand. We are in close contact, and we had discussed what could be the goals and the overriding objectives, and we come together, and it's a very nice cooperation that is a patient organization. Next slide. The name 8-in-1 stroke, it remembers on the German fairy tale about the little brave tailor, and he is a hero because he makes 7 in 1 stroke, and we just want to do one more, one better. And so the 8 is because we are including 8 gangliosidosis in the registry, and also we use in this natural history study a new score with 8 items, and moreover the 8 is called in Germany Acht. And Acht is achten, ein altes old German word, and it has several meanings, and one of them is watch for early symptoms. And another meaning of achten is esteem patients. And that comes all together, that gives the study the name 8-in-1 stroke. Next slide. So I like to present the background, the aims, and the research questions of the study, show a little bit about methods, how is this study conducted and how is this study structured. And the methods are always ping-ponging with the results, and in the end, some discussion and conclusion slides. Next slide. So the overriding objectives of the 8-in-1 natural history study is first, knowledge, understanding gangliosidosis better. Second, awareness, making gangliosidosis known to the public, and participation. We actively involve patients in process and planning. And last but not least, level of access to drug trials through natural history data. So, next slide. The research questions. What was investigated in the study? The 8-in-1 score was newly developed to describe severity of the disease. And the research question is, does the 8-in-1 score reflect severity of the disease? A second question is, how is the disease noticed? From the parents, from the doctors? How do they detect? And we ask every family, what does a delayed diagnosis mean for them? And the third research question is, how does the course of the disease develop? What about progression? What are differences in subtypes, and what are differences and what are the commons in these disorders? Next slide. The study process, a very easy plan, a baseline investigation with medical history, physical exam, the 8-in-1 score, the Wineland, Ruben had explained it, Sarah also, the 9-hole PEC test also explained before. And then that's new, WHO does. It's an instrument developed from the WHO to quantify limitations and handicaps. And then committed medications were documented, and events such as seizures or hospitalizations. And that was done every year. Only for the Wineland score, we decided to make the baseline investigation and the first one, and then decided afterwards if in 1 year there is a significant difference, and may later on we make it less often. Next slide. At this time, in the middle of December, we have enrolled 29 patients, and 3 further patients were dated for enrolling. And the most of the patients here in the middle are Tay-Sachs disease patients. But also frequent, we have GM1 gangliosidosis and Sandhoff disease patients. We have 2 patients with GM2 activator deficiency, but the other 3 gangliosidosis, Morquio B, C. alidosis, and the B variant, B1 variant, we have not included so far. 1 patient with C. alidosis asked for participation, but so far not included. Next slide. What about the 8-in-1 score? What's that? That's a score adapted from scores used in NPC and NCL, other lysosomal neurodegenerative disorders. And we added to this because we felt that it's of value to add participation and independence. Each of the 8 items were rated from 0 to 4. O means no restrictions in this item at all, and 4 means loss of function or complete, most severe handicapped in this item. And 1, 2, and 3 are in between 0 and 4. Next slide. And this slide shows a comparison how the parents and how the doctor is rating the 18-1 score. The 18-1 score, 8 items, 4 points, so it's from 0 to 32. And you see there is a high agreement between the investigator rater and the parents or the caregiver rating. There's a high agreement, but there is a trend that parents rate their children better than doctors. Yeah, that could be one point is for sure. Parents know better and see maybe at home some better things than in the hospital the doctor is seeing. But also we recognize that patients have difficulties to understand the definition of epilepsy and the rating there. And especially these ratings where the parents have more than 2 points better than the doctor, it's explained by differences in epilepsy rating. Next slide. In the second step to analyze the data, the baseline data of 18-1, we compared the 18-1 score with other tools. You have already heard about the Vineland adaptive behavior scale. It's a telephone interview of the parents and the caregivers. The SARA score is explained in 9-hole practice, and WHO as I also explained before. And the next slide shows the results in the comparison. Here in this slide, on the left, you see the comparison of 18-1 and the Vineland scale. It looks in the good performing Vineland patients, there was a correlation, but here on the end, in the bad performing patients, there is no discrimination, no focus, so you could not differentiate severe and most severe patients. Here, the correlation between 18-1 and motor function in the SARA score, it looks like a good correlation, but the limitation is only a low number of patients who could perform because of age or because of disease severity this test. It's also a good correlation to WHO DAS, to the grading of limitations. It's a fine correlation, but that's only validated in adults, and we have not done it in children. The correlation with the 9-hole PEC test, it's limited. There seems to be only a few patients who can perform the test, and then especially in the good end, there seem to be a plateau or no the other factors which influence the dexterity in these patients. Next slide. This is an analysis looking for the 18-1 score related to age And you see here this quarters. These are the infantile patients, and in a low age they have a high number and a 18-1 score. It's this group of patients is different to the late infantile patients, but in this group here are the GM2 patients, and here are the GM1 patients with late infantile. So there seems to be a difference. And also here, between the juvenile and the adult group, there seems to be a differentiation, but in two patients, two juvenile patients look like they have the same score as adult patient at a given age. Next slide. It's a little bit small, but it shows you for the four phenotypes I have characterized the diagnostic delay. Diagnostic delay means the time between first symptom and diagnosis. And here in the first graph, these are the infantile patients. It looks, compared to others, is only a mild diagnostic delay. But in reality, 1 year, 9 months to 1 year diagnostic delay is in infantile patients quite a lot. It is unacceptable, this diagnostic delay. In the late infantile group, there is a diagnostic delay in median by 2.5 years, and for adult and juvenile patients, the median is about 5.5 years in both groups. Next slide. This on the first view, it looks like a very busy slide, but I let you through. And every bar shows a patient, and the bar has different colors, and every color means a phase of the disease. The first phase, the green one, is the symptom-free phase. Then development delay, initial phase of regression, assisted walking, no locomotion, and minimal or no motor function at all, at the most severe. And this gives you the information how it looks like, and it's very important to recognize that the greater the symptom-free interval, the slower the disease progresses. And the second, what we find here, that late infantile GM1 patients and GM2 patients differ. So the GM2 patients, they progress after starting with regression very quickly, and the late infantile GM1 patients, the assisted walking and initial phase of regression is much higher than in GM1 patients. Next slide. We have shown this in these differences between GM1 and GM2 patients in another way. That's with the technique of Kaplan-Meier curves. It gives you the age. Here, this slide gives you the age of the first motor symptom in GM1 and GM2 patients. So the first symptom is very similar in these patients. That's by definition. So, but the loss of motor function, so no locomotion at all, it's quite 10 years later in GM1 than in GM2. Next slide. I must say we have learned a lot in this study from parents. They give us the information about first symptoms, about progression, and about burden of disease. Thanks to all families who participate. Next slide. And what's very important, what I learned from these results, that we have to do a paradigm shift. I don't know if this is only a German problem. German doctors, they learn when to think about gangliosidosis in the time of loss of abilities, of macrocephalus, and cerebral spot. And that's written in the textbooks. But we have to teach the doctors that all development disorders have to screen for gangliosidosis. And there are in the late onset forms and also in some early signs like startle reactions in the infantiles and cerebellar signs and leg weakness, and maybe also a little bit later psychiatric symptoms in the late onset forms. And also important, if the ophthalmologist find no cherry red spot, it does not exclude gangliosidosis. That's what I have heard from German doctors always. Next slide. Limitations. We had so far no patients for 4 diseases yet. We had not automated real-time database analysis, and so far only presented to public at the annual German Pediatric Conference. We will have a manuscript submitted in this month, and so far no data on progression, and disease patients could not include it so far. We have amended this to the protocol and wait for approval for this. Next slide. The strengths, 29 patients for eye disease. It's a large number. It's a first publication with quantification of late infantile course in GM2 patients. It is a good and simple tool to assess severity of overall disease. And we learned a lot from comparing this aid in this time 4 diseases. It's company independent, and it gives us a lot of information about the first symptoms, what we have to look for. And we got first impressions about the course of the disease and the evolution of the disease. Next slide. I have to thank Professor Markus Ries. He is the one co-investigator from the University of Heidelberg, is a good friend, and it was wonderful that he joined the team of the Sphinx. Next slide. That's the team of the Sphinx who had done the study, including Laila Arash and Michael Beck. Thanks a lot.
Sorry, my volume was off. Thank you, Dr. Mengel. I had myself on mute. Thanks for joining us. Great presentation. We're running slightly behind on time, so I'm going to hand over straight to our next speakers just so we can catch up. But if there's any questions from anyone, please put them into the chat and we can direct them over to Dr. Mengel for him to answer. So thank you again. I'm now going to be bringing in the team from Tay-Sachs. So here's Jen, Dr. Schersch. Great to see you guys. I now have your presentation. Because we're running behind, I'll let you guys kind of kick off, introduce yourself, and then run through your talk. So we're really looking forward to hearing it.
Great. Well, hopefully you can hear me fine.
Yeah, we can. Thank you.
Great. Thank you. And Jen, do you want to say hello as well, just to make sure we're hearing you as well?
Hi, everyone. Nice to meet everyone, and thanks for including us.
Great.
1 second. Let me try something else. Give me 1 second. There we are. I will control them, Jen. It may be easier. So you just say next slide.
Okay, thank you.
Great. Well, why don't I kick off? Nice to see everybody. We're very, very excited to be here, myself and Jen. Hopefully you can see me. I'm actually based in California, and it's a bit dark outside, so I've got my desk light on. Hopefully you can see me fine. But it's a real pleasure to be here. My name's Suyash Prasad. Let's go to the next. This is our legal disclosure statement. We need to have this on every slide. My name's Suyash Prasad. I'm the Chief Medical Officer and Head of Research and Development at Tay-Sachs Gene Therapies. I trained as a pediatric intensive care specialist, so I'm actually very used to looking after children with severe progressive metabolic disease, especially towards the end stages of life. And I moved into industry about 20 years ago and clinical research about 20 years ago. And my career in industry has really been focused on rare and severe metabolic and neurological disease. And I find myself here at Tay-Sachs, and I'll tell you a little bit about Tay-Sachs in a moment, but we're a company that's focused on developing gene therapies for rare neurological diseases. Let me pause there, and I'll ask Jen to say hello as well.
Hi, I'm Jennifer Van Houten, and I'm Associate Director of Patient Advocacy for the Tay-Sachs and Sandhoff program, GM2 gangliosidosis. I actually have a little bit unique experience as I had 2 children with a different lysosomal storage disease and worked quite intensely in caring for them, as well as being active in research for their disease. And so I'm happy to be able to bring a unique perspective to Tay-Sachs and be able to help the team understand this lysosomal storage disease.
Great, thank you, Jen, and you certainly do bring that unique perspective. So we're very happy to have you here. Okay, let me tell you a bit about Tay-Sachs. So Tay-Sachs, as I say, we're actually headquartered in Dallas. We have some laboratories in South San Francisco, and we're building out more labs and our manufacturing for the gene therapy in North Carolina. So we're a US-based company, but we run our clinical trials globally. And we have our regulatory interactions globally. Our focus is really what we call AAV9 serotype gene therapy. And this is AAV9 is a shell, it's the capsid that surrounds the actual genetic information. And it's the serotype that has the most clinical experience. You may well have heard of a disease called spinal muscular atrophy, and the fact that that's an approved gene therapy, and there's about 1,500 patients on that gene therapy currently worldwide, and that was AAV9. And many of us at Tay-Sachs actually worked on that program at previous companies. We also have a partnership with UT Southwestern. It's a university in Dallas, and in particular with the lead gene therapy researcher and the division chief of pediatric neurology. So the lead gene therapy researcher there is a gentleman called Stephen Gray, who's a very well-known, well-established gene therapy researcher, and the head of pediatric neurology is Burj Manassian, very, very experienced and very really extensive background in metabolic and neurodevelopmental aspects of pediatrics. Next slide, please. So We have a platform approach to treating rare and severe neurological disease. And so everything we do is the AV9 capsid. So we don't change capsids. You may have heard of different serotypes, AV8, AV2, AV5. We stick to AV9 because we know how to make it and we know how to scale it. We also use a manufacturing process known as HEK 293 suspension. Okay, HEK stands for human embryonic kidney, and it's a manufacturing process for gene therapy that once again we think is proven, and we like it and we know how to make it. And we give drug intrathecally, so we target the brain and the spinal cord. So what this means is we pop a needle into the lower spine of the child and take out some drops of cerebrospinal fluid and then inject the gene therapy drug into the CSF intrathecally. Many of you will know what a lumbar puncture tap is. After the administration of the gene therapy, which is usually 8 to 10 mils of drug, of fluid, we lay the child on their back, head down for about an hour, and allow the drug to bathe the brain and spinal cord where it can then enter the brain cells. So we have that platform approach across all our programs. We actually have a number of programs, several lysosomal storage disorders, other neurodevelopmental diseases, but our GM2 gangliosidosis is the studies enroll the second highest number of patients. So GAN is our lead program, and then second to that is the GM2 in terms of advancing forward through the clinical trial stages. Next slide, please. Once again, just to reiterate our partnership with the UT Southwestern group, and the vector design for the GM2 gangliosidosis program was performed by Stephen Gray, who you see in the bottom half, in partnership with Jagdeep Walia, based up at Queen's University in Canada. And it's actually Jagdeep and his partner, who's a pediatric intensivist up there, Anupam Sehgal, who are running the clinical trial currently. Next slide, please. And let's jump to the next slide. So I'm just going to talk now specifically about GM2 gangliosidosis. And we have ascribed our Tay-Sachs number, Tay-Sachs 101, to this particular program. This is the general approach to a gene therapy, or even a drug, a non-gene therapy drug in terms of development. There's a discovery phase where someone has a good scientific idea and they think about it and design a construct or design an entity and then start doing some lab work. It then moves to preclinical research. Preclinical essentially means animal studies. So you then start doing some animal studies initially in mice, then in the mice model, then occasionally in larger animal models also. Once you've met the hurdles of demonstrating safety and efficacy in the animal model, you then move to clinical research, and then you run the clinical trial in a smallish number of patients. And if you're able to demonstrate safety and efficacy in that group of patients, you then move into filing the data with the regulators and hopefully obtaining an approval where you can then commercialize the drug. So for the GM2 program, we're in this green bucket here. So we've started the clinical trial, patients have been dosed. We haven't shared any data from that study as yet, but we're looking forward to seeing what the data show. So we've been through the discovery phase, we've done the preclinical research. There's a little bit of additional preclinical research that needs to be done in parallel, and now we're going through the clinical research phase. Next slide, please. I won't talk to you in too much detail about the disease. I'm sure you all know the disease very, very well, but there's a nuance to the disease. There's an interesting aspect to this particular condition that makes the gene therapy approach a little more challenging. And that is that the enzyme that's missing, beta-hexosaminidase A, is formed of two subunits. An alpha subunit that is coded for by the hex A gene, and the beta subunit that's coded for by the hex B gene. So of course, a mutation in the hex A gene gives you Tay-Sachs disease. A mutation in the hex B gene gives you Sandhoff disease. Essentially, from a gene therapy approach, we're actually treating both diseases with the same construct. And we're able to do this because we include both the hex A DNA, the hex A gene, and the hex B gene in the construct itself. So if we go to the next slide, you can see on this next slide, this is a picture of the construct. And I use the term construct really to show you the molecular structure of the gene therapy. So you can see, if you look at the diagram on the right, in the light blue, you've got the H hex B, that's the human hex B gene, and then you've got the HXA, the human hex A gene. Now, luckily, both genes are relatively small, so they can both fit inside the AAV9 capsid, which is the shell. And you can see that represented diagrammatically at the top of the graph. Now, you see in the middle between the two genes, there's this orange box, and it says P2A. This is a peptide linker. It's a small strip of protein that joins the two together, the two strips of DNA, within the actual shell, okay? The shell being the capsid. And you can see next to the HXB, in the darker blue, you've got something called a CAG. Now, the CAG is what's called a promoter. So this is the engine that encourages expression of the DNA that then gets translated to mRNA in the cell, and then that gets translated to protein in the cell, specifically the alpha unit and the subunit, and then the alpha unit and the subunit can join to form the functioning heterodimer within the cell itself. So it's a somewhat unusual design from a gene therapy perspective. This is what we call a bicistronic vector, because there's two genes wrapped up in one capsid, and the two genes are linked by a peptide linker. And it's actually the first time a bicistronic vector has ever been used in a clinical trial. So we're quite excited about this approach. So that's the construct, and that's a little bit of the molecular biology. And a lot of this information is published, and so you can read the clinical papers. You can see actually the reference at the bottom of this particular slide. I've got two slides of the animal data. I've told you already that the preclinical research has been conducted, but if we go to the next slide, you can see here, these are knockout mice. So this is actually the Sandhoff knockout mouse model, and this is a survival curve. So what this is showing is that if you look at the y-axis, that gives you number of mice or percentage of mice that survive. So if this was just healthy mice, what you'd see is a flat line at 100% until they are about 18 months old, when the mice would start to die. Now, the blue line is the knockout mouse. So this is the disease model mouse that's untreated. Vehicle means you give them placebo. So you can see here that the blue line shows that most of these mice die around about 16 or 17 weeks of age. So these are mice with Sandhoff disease. Now, the black line, the green line, and the yellow line are where we give increasing doses of the gene therapy construct. Okay, so low dose, medium dose, and high dose. So you can see the black line, being given the low dose of the gene therapy, increases survival by a little bit. The average survival is about 26, 27 weeks. When you get to the medium dose, the average survival is about 42 weeks. When you go up to the high dose, the yellow line, you see the average survival extends quite significantly. So this is showing a treatment benefit that is dose responsive in the knockout mouse model. So essentially, this was what we call a very positive, clear animal pharmacology study. And this was the study that demonstrated to the regulators, okay, this drug, this gene therapy construct has a good chance of working in the human situation as well. Next slide, please. Of course, that was survival. And what we're showing here is a biomarker. So there's a couple of biomarkers you can think of in this particular condition. You can look at hex A levels, so beta-hexosaminidase A levels. You look at that in the serum, i.e. the blood, or also the cerebrospinal fluid. There's a lot less information looking at hex A levels in the CSF, but we've actually done some background work, so we think this might be a good biomarker of what we might expect clinically. You can also look at the accumulation of GM2 ganglioside, i.e. the substrate that should be broken down by the beta-hexosaminidase A. And what you see here is in the knockout mouse model treated with vehicle or placebo. That's the light blue line. You see plenty of substrate. And as you go up in dose, the black line, the green line, the yellow line, you can see you're removing the accumulated substrate. So this is showing improving pathophysiology which fits in very well with the survival curve you see previously. So this was our mass pharmacology study that was persuasive to the regulators to allow us to move into clinical trials. We, of course, also did a toxicology study where we overdose animals with large amounts of drug. And that was actually a very positive study as well. And that combination of the pharmacology study showing drug efficacy and the toxicology study showing safety demonstrated the regulators we had a nice positive window of therapeutic efficacy, which allowed us to then move into clinical trials. And as I said, we dosed the first patient earlier this year, and we're hoping to share data from that in the next few weeks. Next slide, please. And I'll now hand over to Jen, who's going to talk a little bit about our interaction with patients. And it's particularly important because that interaction with you all really helps us have our discussions with regulators and actually informed our clinical trial design. So let me hand over to Jen.
Thank you, Suyash. So next slide, Dan. Thank you. This is our patients that are describing our patients that are really centered to the work that we do at Tay-Sachs, our patient guiding principles. And you can see within collaboration, it's founded on really the basis of collaboration between the partners that Suyash spoke earlier about with UT Southwestern, now Tay-Sachs, and then our patient advocacy groups that we work with. And we want to make sure that we can, focus on the coordination of the patients and their families and make sure that we can also connect with the clinicians for moving things forward. In transparency, we really strive to be a true partner by providing proactive communication that is timely and informative. And curiosity, here at Tay-Sachs, we work closely with the patient communities and to understand, the patient journey and what is that like on a daily basis. And then also, what are the challenges for patients and their families that are living with Tay-Sachs and Sandhoff disease for this community. Empathy, we lead with patients and their families in our work, and we are committed to provide the compassion, empathy, and care beyond the product itself. And so we strive to pull that through our inner workings at Tay-Sachs. And then respect. Tay-Sachs really honors the privacy of patients and their families by strictly adhering to policies that protect their personal and medical information. Next slide, please. You may have heard this before, but the importance of 3 key groups working together to bring therapies in patients with rare diseases are advocacy, research, and industry. We have strong advocacy and patient communities, especially within the Tay-Sachs and Sandhoff community, to help us advocate for the patients and raise awareness of the disease and making sure that they receive the attention that they deserve. The ties to academic, which is UT Southwestern and Queens, and the clinical research centers ensure discoveries in science will move forward. And then industry provides support and the resources and expertise. Suyash mentioned some of the team's expertise in the first commercial product for another rare disease. And this expertise helps navigate through the regulatory and the commercial pathway. And for me, this partnership has never been more apparent as we are working side by side with UT Southwestern and these incredible patient groups in the Sandhoff and Tay-Sachs communities every step of the way. We truly believe that embracing this integrated approach will help us be able to get medicines safely and effectively to the patients in the best way possible. Next slide. With engaging in the patient community, it's, I've mentioned it's central to our clinical development work, and really this partnership with the patient advocacy groups, including CATS Foundation, we've worked with some other European Tay-Sachs and Sandhoff consortium partners, it's critical to be able to move the GM2 program forward for Tay-Sachs and Sandhoff. I'd like to start with some ways that we've been able to work with your community. In the summer of 2020, we held a work group with several Tay-Sachs and Sandhoff parents that helped us understand the unmet needs of what the kids and their families were going through. And this helped us understand areas of educational needs for the families and clinicians about our gene therapy program. With this information, and I'll share a little bit about it later, we developed clinical trials to develop based on the patient and family insights. These interactions can help us bring awareness to the clinical trials and make enrollment more efficient. Together, we can work to advance new therapies for Tay-Sachs and Sandhoff disease. Next slide, please. This is a overarching view of how we work with the caregivers on co-creating a gene therapy clinical trial. Our objective was to make sure that we can gather the perspective of infantile GM2 gangliosidosis patient experience. And we then were able to inform the development of a clinical trial protocol and supporting materials. The methodology for this program is pretty simple, and it's actually ongoing throughout about a month's time. We have a recruitment process where we worked closely with NTSAD in the United States to be able to find patients, the caregivers of the patients, to be able to join in on the program. And we conducted an online survey with these patients once they were consented. And then within the online survey, they also were able to have an online forum where they could, meet each other. Often they do know each other, but sometimes they don't. And they can talk about different, parts of their day or the symptoms that they might be seeing in their children or have seen, and work together through some of these different conversations. And then we also did a content analysis, a survey form and output that provided information to our internal team. And we hosted about a 2-hour video focus group. With COVID, it's been very, very different in doing these focus groups. We have done them in my past experience in person over the course of about a 6-hour day, but this is broken up a little bit because of, the time spent on Zoom and in front of a camera is a little bit more, it's just a different experience than being in person. So we had a nice discussion with these caregivers. And then from there, we were able to pull together a data analysis and report to be able to share key learnings within Tay-Sachs and Sandhoff disease. So what do we learn? Sorry, Dan, next slide. So here are some of the pieces that we learned about in the work group with the Tay-Sachs and Sandhoff caregivers. We learned about their journey to diagnosis. And several of the parents saw missed milestones, like not sitting up or maybe sitting up at one point and then regressing a little bit. And then they also noticed low muscle tone about 4 to 6 months of age. And again, this is a small snapshot of the caregivers that we had a conversation with. So this is not an overarching Tay-Sachs and Sandhoff perspective, but we do like to have these type of conversations with the parents. Some of the parents also reported that their pediatrician recommended physical therapy and occupational therapy. And then when they didn't see that they were getting better or progressing with PT and OT, there was a neurology referral that was made to look into a genetic cause. And about, on average, it was 9 months when the parents were able to get this connection with a genetic, with a neurologist. The average age of diagnosis was 10 months, and within the group that we worked with, it ranged between 2 months and 16 months. And then, as I mentioned before, we like to learn about the most challenging symptoms. These are really hard for the families to be able to manage and cope with, and we want to make sure that we can understand the disease so that we can develop the most appropriate clinical trial. And some of the most challenging symptoms that we learned about were swallowing difficulties, and I mentioned the low muscle tone, decline in motor skills, contractures or muscle spasms, and then sleep issues, and then seizures. Dan, next slide, please. So as I mentioned, the Tay-Sachs has incorporated these caregiver experiences because of this work group and was able to help prioritize and share what the needs are for the families in the study design. Some of the pieces that we were able to incorporate were the presence or absence of severity and frequency of seizures, age-appropriate quality of life, and what that means to the families, and then physical, emotional, social, and cognitive functioning, as well as motor neurodevelopmental and cognitive status of their child. And then really this is one thing that we found across with most of the families is that communication and social interaction within their home communities were also challenging for the family when they were caring for a loved one. So now I will turn it back to Suyash to talk about the clinical trial update.
Thank you very much, Jen. And I just want to re-emphasize the fact that it's really vitally important for us to hear from all of you as we think about designing the clinical trial, and actually in the context of regulatory interaction as well. We've had several discussions with regulators from all over the world on GM2 and indeed our other programs, and it's always important to weave in the patient experience. So as Jen says, some of the endpoints we've chosen for the clinical trial really have been specifically chosen based on your feedback. For example, we've got Jen mentioned the fact that communication and socialization is really important. So we've included a scale called the ORCA, the Observer Reported Communication Ability Scale, which is something that you as parents will rate. And of course, we're looking at other parameters that you've told us are important, such as the acquisition of milestones, developmental milestones, the loss of developmental milestones, seizures, how frequent the seizures are, how severe they are, how long they last, and several other quality of life measures, as well as clinical endpoints. Now, in terms of the study update, the name of our gene therapy drug is TSHA-101, TSHA-101, and we are running it in a single-arm open-label study in Canada currently. The intent will be to open it up into different parts of the world, but we're able to get the study open up through the regulators in Canada sooner than other places, so we started dosing patients up there. And part of that is because the co-designer of the construct, Dr. Jagdeep Walia, is based up in Canada. So the trial is currently enrolling children. We started off with a fairly narrow selection criteria, so it's children aged 15 months or younger. My guess is that as the next year progresses, we may widen that or broaden it to include older children. But to start off with, we really wanted to focus on the really critically unwell children at a young age where we thought there may be an opportunity of stabilization of the disease process. So that's the current inclusion criteria. And as I say, the construct treats both Tay-Sachs and Sandhoff because we include DNA for both the Hex A and the Hex B subunits. And it's an international study. Even though the study's been run in Canada, we are very happy to transport patients from all over the world. We've transported patients from South America, from the Middle East, from Europe, and we cover all the costs for that. You know, if you're somewhere in the world and would like to take part in the study, please do reach out to the team at Queen's University, or you can reach out to Tay-Sachs, and through us we can connect you. And if your child is suitable, and you know, obviously you'll need to be in the queue in the line, but wherever you are in the world, we'd generally be happy to transport you and cover all the costs for that. In terms of data that are coming out, I'd always planned to share data by the end of the year. Likely it's going to be the first week of January. We don't really want to issue a press release or talk about data whilst everyone's on holiday, but we will be sharing that in the first week or the second week of January. And it'll be preliminary data on a small number of patients that have been dosed. It'll generally be clinical safety data, how have the children tolerated the drug from a safety perspective, but we anticipate also being able to share biomarker data, specifically the beta-hexosaminidase A levels in the serum and in the CSF also. Next slide, please. I put this slide on because we actually work on a number of programs, and I think it's important to make this point because we're moving several programs forward in parallel, but you can learn from one program to inform the others. Once again, everything is AAV9 capsid, everything we do is intrathecally delivered, everything is neurological, neurodevelopmental, neurodegenerative disease of children. So by the end of this year, we'll be sharing data from the high-dose cohort from our giant cell neuropathy study. This is another neurodegenerative disease. 14 patients are dosed on this program already. We'll be sharing the biomarker data from the GM2 program and the safety data from our CLN7 program. This is a form of Batten disease, and we'll be sharing that a bit later on in the next few weeks. By the end of next year, we'll be sharing biomarker data from the CLN1 program, which we anticipate being able to dose patients probably Q1 next year, and also clinical data from our RET program. And once again, we anticipate dosing patients in the first half of next year. And of course, we'll be sharing more updates on GM2 as the year progresses. So we have lots and lots of things, lots and lots of exciting data to look forward to. Next slide, please. In summary, our gene therapy is under investigation. It's in clinical trials currently up in Canada. We're delivering both the DNA for the HexA and the HexB mutations, so treating both Sandhoff disease and Tay-Sachs disease. We have very positive animal data which supported the opening of the clinical trial. We've been engaging with you all to help inform the clinical trial design, and we want to continue to engage with you all on an ongoing basis. And I look forward to sharing data in the next few weeks with you. On that note, I'll end, and if we have time, I can take questions, or if we don't have time, we can move on. Dan, I'll leave it to you.
Oh, that's fantastic. Really great to hear the update and all the work you've been doing. Obviously, many of us have been involved in the advocacy side, and it's really encouraging and exciting to have you guys kind of bring us into the conversation and help lead on things. I think this, as I said before, approach is going to be very, very important as we move forward. So we're running slightly behind. So what I'm going to propose is we take a quick 10-minute break for now. If anyone has any questions, please just save those up and we can ask them in the panel discussion later today, or we can message them over to us and we can make sure they go to the right team to answer. So we'll have a quick 10-minute break. We'll be back at, so it's 3:50 my time to restart the next session, which will be Jennifer from SCIO Gene Therapies. So thank you, everyone, and I'll see you in 10 minutes.
Thanks, Dan.
Hello, everyone again. Hello, Jennifer. Nice to see you.
Good to see you as well.
So we are continuing with the last part of our conference. We have with us, we are still in the other side of the ocean in the US with Jennifer Papadakis. She is Vice President of Medical Affairs at CEO Gene Therapies. As part of her role, she meets with scientific experts and patient organizations to get feedback for the GM2 and GM2 gangliosidosis clinical programs, educational needs, and diagnosis challenge. So she is going to give us a brief talk about CEO's GM2 gene therapy for the treatment of GM2 gangliosidosis. So thank you very much for joining us, Jennifer.
Oh, thank you, Bia, for the kind introduction and for the invitation to be here today. For those of you that may not be familiar with SIO gene therapies, we previously were named Axivant and changed our name about a year ago. But we have two clinical programs, both gene therapy programs for rare diseases ongoing. One for GM2 gangliosidosis, which is being conducted with Dr. Florian Eichler at Mass General Hospital and Terry Flott at University of Massachusetts for Tay-Sachs and Sandhoff disease. We also have another program for GM1 gangliosidosis, which is being conducted with Dr. Cynthia Tift at the NIH. But today I'm very excited to be sharing a little bit more about our GM2 gene therapy. The technical name is AXO-AAV GM2, but that's a mouthful for me, so I'll just be referring to it as GM2 gene therapy for this presentation. So if we could have the next slide, please. Today I'll be covering a little bit more about our GM2 gene therapy, design of our clinical trial. I'll provide a status update of the trial and then answer any questions that you may have. Now, I know other speakers have covered what the cause of GM2 gangliosidosis is, so I'll really just utilize this slide to explain how our GM2 gene therapy is believed to work. So Tay-Sachs and Sandhoff disease is caused by mutations in either the HEX-A or HEX-B genes. And without working copies of these genes, there's low levels of HEX-A enzyme, which then leads to accumulation of toxic GM2 storage materials in neurons in the brain and spinal cord. This then leads to the symptoms that we all know too well in infantile and juvenile patients. With our SIO's GM2 gene therapy, we provide or deliver working copies of the HEX-A and HEX-B genes, which allows cells to produce HEX-A enzyme, which then begin to break down the toxic GM2 storage materials in the cells and potentially can either slow or stop disease progression. Next slide. So I'll share a little bit more about SIO's GM2 gene therapy on this slide and what it is. So our gene therapy is delivered in a liquid form, and it contains working copies of the HEX-A and HEX-B genes, as well as a harmless non-disease-causing adeno-associated virus, or AAV, which is used to deliver the genes to cells within the brain and spinal cord. Now, we use a virus to deliver the cells because we know that viruses are very good at entering cells. So when I refer to our AXO-AAV GM2 or our GM2 gene therapy, I'm really referring to the HEX-A and HEX-B genes packaged in the adeno-associated virus. And for SIO's GM2 gene therapy, we deliver the HEX-A and HEX-B genes in separate of what we call vectors in a 1-to-1 ratio. Next slide. So our GM2 gene therapy has been studied in a variety of animal models. mouse models, a cat model of Sandhoff disease, as well as a sheep model of Tay-Sachs disease. But the majority of the studies have been in a cat model of Sandhoff disease because it's more similar to disease progression in patients. So these studies have looked at a variety of ways to administer the GM2 gene therapy. And as you can see in the table, studies have looked at delivery to the deep brain or thalamus. And the thalamus is known to be a very important part of the brain that's affected by GM2 gangliosidosis. They looked at delivery to through the spinal fluid, and then delivery through both the deep brain and the spinal fluid. They then looked at a variety of different measurements, but one of the measurements they looked at was the ability of the GM2 gene therapy to clear out these toxic GM2 storage materials. And in the table, we show by the darker check marks where there was complete clearance of these toxic materials. The lighter colored check mark shows partial clearance. And when there's no check mark, that means there wasn't any clearance of the toxic storage materials. So as you can see from what they found in these studies was that delivery to both the deep brain or thalamus and spinal fluid was needed in order to get complete clearance in the thalamus, the cortex or the covering of the brain, the cerebellum, and the spinal cord. So for our clinical trials, based on these animal models, we decided to administer our GM2 gene therapy through both the thalamus and spinal fluid. And I'll share a little bit more details about that procedure of administration on the next slide. So this slide shows the administration procedure used in our GM2 clinical trial. There is a dual administration procedure. In the first step, we administer gene therapy to both sides of the brain into the thalamus. And on the second day, we use a microcatheter, which is inserted into the intrathecal space along the spinal cord. And we thread the catheter into the cavity in the back of the brain called the cisterna magna, as well as threaded into the lower part of the spinal cord. And really the goal of this administration method is to be able to get good and broad distribution of the GM2 gene therapy across all parts of the brain and spinal cord. So as I mentioned, we're currently studying this in a clinical trial, and on the next slide, I'll share a little bit more about the design of that clinical trial. So our clinical trial is currently in a dose escalation study, and the goal of this study is to assess the safety as well as identify the optimal dose of gene therapy. So we're looking at 4 different dose levels. Starting with group 1, we have a starting dose, then progress to the low dose, the mid dose, and the high dose. Now, this starting dose that we're using is anticipated to provide clinical benefit, but we also wanted to increase the dose to see if we could get any additional clinical benefit. But when we do that, we also need to look very closely at the safety to make sure that we're not seeing any safety concerns as we increase the dose. So we follow safety very closely in this trial. We have an independent data safety monitoring committee which consists of disease experts, gene therapy experts, as well as a pediatric neurosurgeon. So this group, every time we dose a patient, we send a month of follow-up safety data to this committee. They assess it and then determine if we can dose an additional patient or move to the next dose. So where we are currently with this trial is we've completed dosing of both the starting dose and low dose, and then we will be continuing dosing with the additional dose levels. After we complete the dosing, we'll take all of the learnings from this trial and determine the optimal and safe dose. We'll then be conducting a second trial, an efficacy and safety trial, really with the goal of assessing how well that optimal dose works. Results from the safety and efficacy study will then be submitted to regulatory authorities. So this is the FDA in the US, the EMA in Europe. And if we receive approval at that point, then we can market the product more broadly. Just a few things to note with our trial. We are including both infantile and juvenile onset patients. And for each new dose level, the first patient enrolled is a juvenile patient. So if we go to the next slide, I'll share a little bit more about the key eligibility criteria and endpoints for the trial. So as I mentioned, we're including both infantile onset and juvenile onset patients, and there is an age criteria. So for infantile onset, patients must be between 6 and 20 months of age at the time of dosing. And for juvenile onset, patients must be between 2 to 12 years of age at the time of dosing. Patients also have to have a genetic confirmation of either Tay-Sachs or Sandhoff disease. And then our investigators look at other factors, looking at various functional criteria as well as safety criteria. And a more detailed list of all of the eligibility criteria can be found on the clinicaltrials.gov website. Looking at the key endpoints, as I mentioned before, the main endpoint that we're looking at in this trial is safety. But we are looking at other signs and symptoms of disease. And we work with a caregiver advisory council to give us advice as to which symptoms are really impact the quality of life for patients the most, and which endpoints would be most meaningful to them. And they've been really wonderful in guiding us, not only with our endpoints, but really in how to best communicate this information to a family audience. We're also looking at other endpoints such as change in brain volume through MRI, and looking at biomarkers, including enzyme activity and GM2 storage in both spinal fluid and serum. We could move to the next slide, please. I'll cover a few frequently asked questions we often get from families about the trial, but then I'm happy to answer any additional questions that you have, either at the end of the talk or during the panel discussion. So one of the questions we commonly get are patients from outside the US eligible for this study? As I mentioned before, the study is being conducted at the University of Massachusetts and Mass General Hospital, but we do include patients from outside the US as well as within the US, and have several patients from outside the US enrolled in the trial. We have a service that provides assistance with travel and accommodations, and cover the costs, appropriate costs related to your flights, getting a visa, accommodations, food and travel, and various other expenses that you may incur while at the study site. We're often also asked, what are the risks and benefits of participating in the trial? Well, participation in the trial will definitely help us better understand Tay-Sachs and Sandhoff disease, and may help lead to a future treatment. While we have seen treatment benefits in several different animal models, the treatment benefits in patients is currently unproven, which is why we're doing the study. But we certainly hope to see clinical benefit. And there are also risks associated both with the gene therapy as well as the surgical procedure. And if your child is eligible and they talk to the study site, the investigators can provide all of much more detailed information about the trial risks and benefits to you. If you are interested in having your child participate in the trial, we always encourage you first to discuss the eligibility criteria with your child's physician. And if you're interested in further information, either your child's physician or you may contact our clinical research coordinator at the site, and I've provided her contact information here. So if we could go to the next slide. I just wanted to provide an update as to where we are with the study. We have 4 patients dosed in the trial and have completed dosing, as I mentioned before, of both the starting dose and low dose. We are actively recruiting patients for the additional dose levels through our investigators at the Massachusetts General Hospital and University of Massachusetts. And we expect to share some initial data maybe by mid 2022 at the latest. And I think just one more slide here. I just wanted to take the opportunity to acknowledge all of the patients and families and patient organizations for their participation and support of our studies. Also wanted to acknowledge the investigators and researchers that have really led the way in getting us to the point we are with this clinical trial. As I mentioned, the group at Mass General Hospital, University of Massachusetts Medical Center, Auburn University, and also my colleagues at Gene Therapies. So with that, I'm happy to answer any questions, or I know we're a little delayed on time, so also happy to wait for the panel if that makes more sense, Dan.
Great, thanks, Jennifer. Fascinating talk as always, and so exciting to see the advancements in this. I remember 10 years ago, you know, when Emily was diagnosed, gene therapy was this pipe dream that people talked about, but you know, we didn't think we'd actually see it this quickly. Maybe it would have taken it quite a lot longer. So it's exciting to see what you guys have been doing, what Tay-Sachs has been doing, and you know, everything's giving an opportunity to our families to, you know, to have a future. So thank you for joining. I think what we'll do is we'll save the questions, if everyone doesn't mind, for the panel. If you post those into the stage, the Q&A or the chat, we'll ask those when the panel starts a bit later today. So thank you very much, Jennifer. We're now going to move on. Thank you. We're now going to move on to our next speaker. So I'm very pleased to welcome Glenn Morrison from Recursion, who's going to be talking about their company and their product that they're starting to develop. So Glenn, I'm going to hand over to you. Here are your slides. Like the others, just the next slide and I'll move them on for you. So thank you.
Excellent. Yes, perfect. Thank you, Dan. So good morning, good afternoon, good evening. My name is Glenn Morrison from Recursion. It is a pleasure and an honor to have the opportunity to introduce you to Recursion and to our efforts in exploring REC 3599 for the treatment of GM2 gangliosidosis. Next slide. This is my obligatory slide on forward-looking statements. And next slide, Dan. My disclosures, nothing to disclose other than I am an employee of Recursion. And next slide. Now, some of you may be familiar with Recursion, but I'd like to take a few minutes to introduce you to the company. Recursion is a publicly traded company based out of Salt Lake City in Utah. The company is a clinical stage biotech with a strong focus on the treatment of rare diseases, including GM2 gangliosidosis. What makes Recursion unique is our approach to drug discovery. Recursion is uniting technology, biology, and chemistry to advance treatments for rare diseases. And to do this, we leverage a platform, the Recursion operating system, to decode biology by utilizing machine learning algorithms to distill a data set that includes hundreds of billions of searchable inferences across biology and chemistry, unconstrained by human bias. Now, our team of over 400 employees conducts almost 2 million experiments per week, which has yielded data on over 500 disease models, with 5 currently advancing to the clinic and another 40 in the pipeline. Next slide. And all of this is with a singular focus to decode biology to radically improve lives. Next slide. So digging a little deeper into Recursion's drug discovery approach, the Recursion operating system spans outputs from target agnostic hits from the 2 million experiments I mentioned earlier, and utilizes a relational database to identify potential drug candidates. Those candidates are then validated through orthogonal functional assays and clinically relevant experiments, and then once validated are optimized for advancing to clinical trials. Next slide. What this allows us to do at scale is radically change and optimize the drug discovery funnel by, one, casting a broader net, two, identifying failures early to rapidly narrow the funnel, and three, to target high potential candidates and accelerate them to the clinic. Next slide. Now, the power of this approach is shown on this slide, which represents one of the broadest, deepest, and most advanced pipelines in tech-enabled drug discovery, including REC 3599, circled in green here, for the treatment of GM2 gangliosidosis. Next slide. Now, of course, I know you're all very familiar with GM2, and I'm sure this is presented over many times over the past days. GM2, of course, is a rare genetic disorder that progressively destroys nerve cells in the brain and spinal cord. Amongst the forms of GM2, infantile onset is the most common, but of course, juvenile and adult onset also occur. Tay-Sachs and Sandhoff are lysosomal storage disorders where the normal activity of lysosomes is disrupted, leading to the accumulation of toxic compounds. Of course, this leads to the buildup of GM2 gangliosides in the lysosomes, and this accumulation leads to that progressive neurological decline, loss of developmental milestones, and unfortunately death in most patients by 3 to 4 years of age. Currently, there are no approved treatments, but as you've heard, there are several exciting developments in gene therapy and also in small molecule treatments. Next slide. Now, using the Recursion OS discovery platform, REC 3599, otherwise known as robuxastorin, was found to be therapeutically active in a disease model of GM2. Now, this is a compound that has an extensive safety database. It was previously developed by Eli Lilly for another indication and has been administered to over 3,000 adults with an unremarkable safety profile. It is a liquid formulation. It's formulated for administration via feeding tube, and importantly, it has properties which predict high exposures in the brain. Next slide. Proof of concept for REC 3599 for the treatment of GM2 was demonstrated with cells from pediatric patients with Tay-Sachs or Sandhoff. And what you see on this slide here is that in the untreated condition, cells from children with Sandhoff or Tay-Sachs have a significant staining and accumulation of GM2, seen in yellow, compared to cells from a typically developing child. And when we treat these cells with REC 359 in the second row on the panel here, we see a significant reduction in GM2 signal close to that of healthy untreated cells. Next slide. Looking at this graphically with healthy cells shown by the black line in the two panels on the right side, you see little to no signal for GM2 accumulation by fluorescence on the left or aggregate count on the right. And in these graphs, cells from patients with Sandhoff are shown by blue and cells from patients with Tay-Sachs by the green line. And you can see in both panels that as the concentration of REC 3599 increases along the x-axis from left to right, the GM2 signal, as shown on the y-axis, decreases. Next slide. Now, building off of this robust signal in patient cells, Recursion has partnered with patient groups and some leading physicians to design for and plan a proof of concept study in patients with GM2, which is set to open next year, with plans to enroll the first patient in the first half of 2022. Eligible patients will enter a dose escalation period, followed by a 52-week maintenance period, in which all patients will receive REC 3599. And as our first study, we are focused on objectives including preservation of milestones, impact on survival, and of course, safety and tolerability of REC 3599 in patients with Tay-Sachs and Sandhoff disease. Now, we expect more information on the development program to be forthcoming in the next several months, and we look forward to initiating our program with REC 3599 in patients with GM2. And next slide. Thank you again for the opportunity to present, and look forward to addressing any questions. My colleague Lisa Boyette will be in the panel in a few minutes.
Hi, Glenn. Thanks for that. Really interesting to hear the fascinating, exciting work you guys have been doing at Recursion. I mean, it's really grown over the last kind of few months. I've been watching you very closely, how you've developed as an organization. And I think this is going to be a really kind of exciting opportunity for us, again, to have another potential therapy that is not so invasive as with the gene therapy, but could complement the other treatments that are actually out, hopefully going to be in the market very soon. And I know we're going to talk a lot more detail on the panel, and I'm sure people have lots of questions. They'll be flooding in. So I think we're going to move on so we can catch up a bit. Thanks very much, Glenn, for joining.
Excellent.
Really appreciate it.
Thank you. Really appreciate it. Thank you.
Thank you. So the final speaker we have joining us today is a very good friend of mine. It is Nina from Pulse Infraframe. So Nina is going to be talking to us a bit more detail about real world data. Nina, maybe you want to do a bit of an introduction to yourself and Pulse as I get your slides up. Is that okay?
Sure, great. So hello, everyone. My name is Nina Liu. I am the Director of Clinical Product Management at Pulse Infraframe. Before getting started, I just want to take a moment to congratulate the Tay-Sachs and Sandhoff communities on all the great work and advancements you have made in these two disease areas. It really is impressive. And I would also like to thank you for providing me with the opportunity to address the topic of how to bring together the different pieces of the puzzle when collecting real world data.
Still working on the slides, Nina. You just carry on talking. Give an intro to Pulse and then I'll get them up.
Okay, great. So let me start with a brief intro. Pulse was founded in 2011 by Dr. Famita Sridhar. She started her career in academia. She is a pharmacist, clinical epidemiologist, methodologist with over 25 years of experience in clinical trials and patient registries. Back when Famita was in academia, she experienced firsthand the barriers and challenges when conducting research in silo, how it slowed down progress in this race to finding better treatment for patients. So it's really for this reason she started Pulse, to shift that paradigm from a silo approach to a collaborative ecosystem that fosters the viewpoints of not only stakeholders such as KOLs and researchers, but to include the patients and advocacy as well as sponsors into the initiative. But in addition to the stakeholders, she thought about the best approach from a technology and data standpoint in order to support this collaborative ecosystem. So really the ultimate goal here is, how do we generate evidence to move the needle for patients living with rare diseases? So our corporate office is in London, Canada. We also have an office in Cambridge, Massachusetts, and Philadelphia. I'm out of the Boston office. We're a global company with operations throughout the world. We currently support over 17 disease areas and span across over 80 medical sites.
They're just processing. Sorry, Nina.
Yeah, no problem.
Maybe it'd be good to talk a bit more about the patient approach that Pulse takes with its product before we hit on to how the real world data is collected. What do you think?
Yeah, great topic. So one thing, we have heard from the market is that patients, a lot of times we ask them to provide data, share data, and it goes into this black hole. And so, we've been working hard to identify technologies and solutions to help engage the patients. One of the things we have implemented and within our Healthy 2.0 platform is really this whole engagement approach of sharing information back to the patients. We have dashboards that help identify where all the patients are coming from. Additionally, we have what we call My Library, which is really a content management system that allows us to share information back to the patient. And I'm going to talk a little bit more about this in the presentation. Okay, great. So So collecting, generating quality real-world data remains a complex problem. There's so much data out there in so many different formats, stored in different systems, and there are also many different solutions in the market with different approaches and technology, which I'm sure is making it very difficult for you as you are researching solutions that will best address and meet your needs. I mean, it's hard for me when I'm conducting research to really understand the capabilities of the different solutions in the market. So when you're thinking about solutions, there are three key things to remember. Most importantly, look for a company that has the technology and scientific expertise. It's the marriage of these two areas that will provide you with the capabilities, the evidence, and the efficiencies you should be looking for in a solution. A solution provider that has the scientific and clinical expertise in-house will work with you to clearly understand the types of data you need, the types of questions you should be asking, the ways in which to phrase a question in order to get the precise answers that you're looking for without influence or bias. Without this expertise, you may end up with a lot of data that's generating a lot of noise instead of signals. The two other areas to consider is the type of data as well as the capabilities you will need to meet your objectives. So I'll talk a little bit more about these two areas throughout the presentation. Next slide. So as I just mentioned in the previous slide, the quality and the types of data you'll need is important to be successful in your initiative. So we have many partners that we're working with who own existing registries with years and years of data, but unfortunately the data is not usable. Some of the factors contributing to this is that the data that they have collected is not structured, rendering low quality, so it's very difficult to query the data. Another reason is that the data that they've collected is narrow. Some solutions out there only collect or support EHR data. EHR data is great, but when it's on its own, it may not provide the EMA or the FDA with the evidence required to demonstrate a meaningful impact for a drug to be approved or for pricing reimbursement. Another area we've touched on already is when data is in silo, it creates inefficiencies associated with that approach. Silo data will not live up to the full potential especially within the rare disease space. So I'll give you an example. I've recently participated in a kickoff meeting that was facilitated by an advocacy group. So they have key opinion leaders who are doing research for this particular condition around the world. And what came out of that session, because they were all presenting their findings from their research, is that for this particular disease, how it is expressed is very different from region to region. And so it was apparent that they haven't been talking to each other, and just through this one-day session where the goal really was to understand their interest in collaborating in this global registry, they realized how much information they are missing by working in silos. Next slide. So real-world data and evidence play an important role throughout the phases of drug development lifecycle. So, and I really think it's incumbent upon us to educate the market to start collecting real-world data as early as preclinical phase through approval, pricing, reimbursement, and beyond. The data needed for approval and pricing reimbursement needs to start as early as phase 2 so that the evidence is there to prove a meaningful impact. And especially for rare diseases, the real world data collected can be used as a comparator arm. And I hear this, and it's really important because many patient advocacy groups or patient advocates have been, really communicating the fact that it is really criminal to give rare disease patients placebo within a clinical trial, especially when a drug has the potential to improve the quality of their life or in some cases save their lives. Next slide. So how do we get started tackling these challenges we've just discussed? The first piece of the puzzle is taking this collaborative approach, defining your strategies and objectives for your registry. What are the goals you're looking to achieve with your registry? What research questions would you like to answer? That sounds simple enough, right? But it can be when you take a holistic approach to understand the needs and requirements of all the stakeholders participating, from the patients, advocacy, KOLs, physicians, and sponsors. So here's an example. We're working with a client right now to understand the feasibility of starting a multi-site, multi-center disease registry. So I've participated in interviewing at least 3 physicians who are viewed as the KOLs in this area. And the first question they always ask is, okay, this is an initiative that is being sponsored by a pharma company. Will there be a conflict? Now, these KOLs have all participated in their clinical trials for this particular product. So what they're really asking is, you know, with the pharma involvement, will they be able to achieve the goals and objectives that they're interested in? The physicians want to publish. They communicated what they really would like to do is improve the quality of the services that they're providing at their clinics. So the underlying goal of the physician and sponsor are very similar, and that is really to deliver better or new treatments to patients. But the specific data requirements for each stakeholder is nuanced. So we need to take this into account. Our approach is to develop a minimal viable data dictionary that includes all the questions and clinical variables that are of interest of all the stakeholder. And this data dictionary needs to be approved by the governance committee and to ensure that the data collected addresses the needs of all of the constituents. And it's also important when you start this process to have a transparent governance structure in place to oversee the compliance and ethical requirements that are met. So typically our governance structure will include an IRB, a steering committee, as well as the scientific advisory board. Next slide. So now that you've defined your strategy and your question, now it's to figure out how to identify the data sources that you will need. Key considerations include the types of data. Do you need medical history? Do you need PRO data, genomics, and so on? Do you need prospective or retrospective data, or both? Are the PROs available to assess the measurable impact that therapy has on the patient so that you can get new guidelines or drug approved? And if there is an existing PRO data that is available, will it meet the EMA or FDA threshold for evidence? One example, we have one of the longest running melanoma registry. And as you can imagine, cancer has been studied for a very long time. And we're starting to see that the PROs that exist right now are dated. It doesn't include the latest treatment options or different approaches. So these are really some of the questions you need to ask, whether or not the data that you're collecting or the sources will help you achieve your use cases. So the takeaway here is identify a solution that is data agnostic or can support the data types that you're interested to achieve your use cases. The use cases may evolve over time. You may want to start with characterization of the disease, the understanding the disease trajectory so that families can better plan, and then moving on to validating endpoints. So identify the data and ensure that the platform can support your evolving initiatives. Next slide. So traditional registries are set up as an island on its own, but imagine a centralized secure infrastructure where you can mine the data across diseases to identify whether it's commonalities, common symptoms, comorbidities, or gene variation. Just think about how much faster we can accelerate research. Our data management infrastructure is set up with common data elements such as demographics, profile data, and there's another layer of that includes the rare disease specific data elements such as biomarkers, labs, and procedures. And then there is also a disease specific data layer. So, when an example of how this can provide efficiencies is we're currently speaking with a pharma partner that has a drug in phase 3. They have done a natural history study. They have done a PRO study. Now they would like to better understand the trajectory of a particular, very specific symptom. Traditionally, they would have done this study individually, but now they see the efficiencies that can be gained by combining the data that they've collected so that the evidence will be there and ready when they're ready for approval or market access. Next slide. So here's just a graphical depiction of a centralized solution. So think about it this way: for a global collaborative platform to be successful requires a supportive foundation, like a house. And like a house, there are many rooms with common areas for all to come together while respecting each other's personal space. So whether you are researching one disease, multiple diseases and their subtypes, one site or multiple site, or involves one country or globally, look for a solution that can organize and structure your data to support the different sources that will be coming in. So we do get this question a lot: who owns the data, right? What I shared about mining data across the platform, it's not a simple thing to achieve. So who owns the data? It's the patient who owns their data. But medical sites and existing registry owners who contribute data will always have access to their own data. So it's really through a consenting process of the patients, as well as data sharing agreements that have been agreed upon by all the constituents in order for us to share the de-identified data across the board for analysis. Next slide. So now let's talk about the importance of data standards. The FDA had recently issued guidelines on standardization of real-world data. They listed several challenges that everyone should be thinking about. One of the challenges I like to highlight and cite is the differences in source data capture regionally and globally, using different standards and terminologies for the representation of the same or similar elements. So that there's a lot of words there, but the slide here illustrates what they're talking about. So albumin is a protein that's produced in the liver. Abnormal levels can indicate a potential liver or kidney condition. So how the test used to test the protein levels is serum albumin. And the concept itself can be labeled very differently throughout the different systems, whether it's EHR system or claims data or existing registry. So in this example, the EHR number 1 is listed as serum albumin. In another EHR system, it could be albumin blood serum in paren. So as you can imagine, with all the different concepts stored into your platform, it's going to be very difficult for researchers to identify this particular cohort for analysis. Next slide. So hence the importance of mapping your data standards to an ontology so that the data is usable and is regulatory ready. So we map our concepts when it's, whether it's entered manually or ingested, it is mapped to the OMAP ontology. So in this particular situation, serum albumin is the standard concept. So you can imagine could be thousands of variables that you're capturing. Standardizing this will simplify the process. It will eliminate some of the additional time and money that you're going to need to spend in order to scrub and clean the data so that it's usable. Next slide. So at the heart of a successful research initiative are the participants. So, and I talked about this a little earlier, we do hear a common theme, and you may have experienced this, but participants contributes their data and then it's radio silence and it's time to, until it's time to collect the data again. I think overall, the market is getting better at this. But to keep participants engaged, we need to keep them in the loop and provide them with the relevant information back to them. And there are several ways that we can do this, including sharing the latest publication, educational materials such as the best nutritional diet for the condition the patient is living with, quarterly newsletters. These are all ways, if you have a centralized platform where everyone is accessing the same interface, then with obviously with permissions, so that you can only see the data that you can see. This is a great way to share information back to the community. So really the point here is participants are equal partners and collaborators in the research initiative and should be recognized and treated so. Last slide. So in summary, there are 3 key points I like to leave with you. One, it is important to start with a strategy, a defined set of goals and objectives, and a clear understanding of the questions you would like to address with your real-world data. Another key point is take a collaborative and holistic approach, right? One lens or one type of data, such as EHR or a silo data registry, may not provide you with the breadth and depth of data that you will need to meet your research or regulatory requirements. Lastly, we know that research is not static. So look for a solution that will grow with you as your research and clinical objectives evolve. This does not mean you need to start with everything on day one. It just means you need a solution that can provide you with the efficiencies and the ability to flex and pivot as you progress with your initiative. You can start with a GM1 registry and then have a solution that can quickly be configured to add additional disease areas or subtypes, such as GM2 as an example. And last slide. So I just shared a lot of data and information, and I might have spoken a little fast, but if you have any questions, please feel free to email me at nliu@pulseinfoframe.com. Again, thank you very much for this opportunity.
Great, thank you, Nina. You know, I'm a big passion for registries and data, as you know, so it's really good for everyone else to hear about the work that Pulse is doing and, you know, what the product can do. And it's really exciting to see, you know, it's how it's been launched in other conditions to make a positive impact on their communities. And it's something that, you know, we're trying to do our registry, but we need to potentially look more bigger and global because there's so many different data sources out there. So it's absolutely fantastic. So actually, we've been able to catch up in a bit more time. So thank you, Nina. You're actually going to stay there because Bea is going to be introducing the next session, which is the panel. So I'm going to bring Bea in, and then I'll start bringing everyone in.
Welcome everyone to this end of the conference. Thank you everyone for joining us. So we are finishing our second day, very interesting talks. And now we are having all these speakers to talk about what does the future hold for the GM2 community. We have with us, of course, Dan, Nina Liu from Pulse InfoFrame. We have just heard her. Gemma Houghton from Tay-Sachs Gene Therapies. Professor Tim Cox, a very old partner from this community, who sure will enlighten us. Jennifer Papadakis from Gene Therapies, and Lisa Boyette from Recursion. Thank you everyone for taking part of this panel. And if you let me, Dan, I will start asking to Tim, what is your first idea when we ask this question, the title of this panel? What does the future hold for the GM2 community? What are your thoughts?
Yes, it's kind of you to ask that big question. I hope you can hear me okay.
Yes,
and I did enjoy the talks from many people I've met before, people I've heard of, so it is very nice.
Thank you for joining. If you let me, Dan, I will start asking to Tim, what is your first idea when we ask of this?
Sorry, it's
Great.
just come back to me.
Okay.
Is a
So
30-second
please,
delay.
we can go ahead. We can go ahead. Sorry.
Are we okay now?
Yes.
Yeah, so sorry. I'll start again. Genuinely, this is an extraordinary community, and there's a community I can see in the panel in front that I've not seen before, all of it. And there's a community that we know from your wonderful consortium in Europe, which is very different from the one in the United States of America. And all are good, doing many things, interrelated. I suppose the overriding feeling I had was of infuriated optimism. In other words, it takes so long. You know, children are dying all the time. Young adults are having their lives destroyed by this. Even older patients are. And it does take an enormous amount of time and effort. But I do see now an army of people. I see the element of competition. I see a rather more realistic approach to things without too much boastfulness, which is very unpleasant when people are trying to raise money. And I see a real world attempt, small molecules, suppressive molecules, supportive means to get engagement, collaborative data collection. It's come on so far that it is expanding exponentially. And I think so much of it looks to have genuine promise. This is not just a talking shop. There are real things happening. There are companies with resources, with enthusiasm, with experience. There are young people, naive, coming in, and there are, the big therapies being explored. So we will know quite a lot, perhaps in a year's time, from some of the gene therapy trials and indeed the small molecules. Lovely to see Ruben again, who and his colleague who really, Cecile, who really did drive through a new therapy for a disease, and it's the only therapy at the moment that's been approved. Still 10 years on, so that's amazing. So it's encouraging. There are many things to say and questions to ask, but overall, for me, in the 10 years I've been with you and Dan and Patricia and so on, and seeing what you've done, I must say there's a huge recruitment and interest in this very tough area. Yeah.
That's great. Thank you. No, thank you, Tim. I thought it would be interesting maybe just to move on to Jen from Tay-Sachs. I mean, we've spoken many times, over the last couple of years about the patient and patient group involvement. How important has that been for Tay-Sachs? I know you mentioned it earlier, but do you see our role, getting more important as the future years go on as your study moves forward?
Yes, I Dan, I do think the patient leaders, the advocacy leaders are very vital to not only the trial design, but the helping the communities understand the clinical pathway and where expectations can be realistic. I know I've mentioned this before. I do feel that the Tay-Sachs and Sandhoff community has done a nice job with setting expectations in terms of clinical trials. So the continual connections with the patient organizations is going to be, it's so important to, I think, companies and to Tay-Sachs to be able to move therapies into and helping the patients understand the different therapeutics. As Professor Cox mentioned, there's multiple different types of therapies out there.
Great, thanks, Jen. I thought it'd be interesting to hear from Lisa. Lisa, thank you for joining us today. Glenn gave a great presentation earlier.
Thank you for having us. We're excited to be here.
No, I just thought it would be quite interesting to get your viewpoint, because you guys are, I would say, relatively new to our disease area, and you've made huge strides in the last couple of years. How do you see it moving forward in general for the GM2 community? Obviously people are very excited because all these companies are now presenting at our conference. Tim was probably I think you were the only medical person at our very first conference.
Yes,
and now we
probably
have
was.
everyone here.
Great.
Should we be excited? Do you think there's a big future for us in the GM2 space?
I think so. I mean, we're the new kids on the block, so we're just getting to know the GM2 community this year. As you heard from Glenn earlier, our program is moving forward and we're doing everything we can to move it forward quickly. Not all patients are the same, right? It's great to see a field grow up around a rare indication like GM2, where in a few years, we would hope to see that physicians have multiple tools in the toolkit so that they can have a multi-layered approach to addressing some of these devastating indications that are impacting families in real time. I really related to what you were saying, Professor Cox, about the time that it takes to get a drug to market. At Recursion, that's something that we are incredibly frustrated by. Dan knows I actually come from the rare disease advocacy world as a family member. And so I feel very fortunate to be at a place where we're having conversations about how to innovate on that timeline. Are there things that we couldn't put in parallel? Are there creative ways to work in this highly regulated space and deliver for patients more quickly? So
Yes.
I think I have every reason to be optimistic, Dan. And I think the way this community functions in such a tightly knit way to generate data that is useful to the various stakeholders who are operating in the field, that is shareable and accessible, is going to make a difference, because the only way the next generation of therapies will improve is if we have more insight into disease biology and how the therapies that we have currently are acting to restore homeostasis in that disease biology.
Could I just add one thing? It's very true. Can I just come back a bit? I think we're quite used to having very direct relations, functional relations, without just talking about it, but actual real relations with, proper relations with disease communities. From my point of view, it started with Gaucher disease, which is massively iconic in the field because of the huge investment, the large amount of money that was made and is still being made, and that huge incentive it's provided within the capitalist system. But the capitalist system gets a bit fragile at times. Things go wrong, but it's worked wonderfully for Gaucher. It worked wonderfully for cystic fibrosis, if you happen to live in a Western country. And it's worked pretty well for some of the hemophilias. But it doesn't work for most of those rare diseases all that well at the moment, unless the therapy is completely transformative. And so you're looking for something that's dramatic. And I'm going to ask you then, do you think that any one therapy for, let's say, infantile, let's start at the tough end, or for juvenile, pretty tough, and they're all tragic. Actually, there's no competition there. Or even for the more adult so-called attenuated forms. Do you think any one therapy is going to do it, honestly, in your heart of hearts?
Well, I'm a scientist. I'm driven by data. I think we have to wait and see what the data show us. I know we're all very excited about gene therapy. I talk with families on a weekly basis, trying to get those studies. If that was the solution for everyone, I think as a rare disease community, we would all rejoice, right? And that,
of course,
remains to be seen.
Yeah. Well, we could come to that. Yeah. But preclinical data in Tay-Sachs that we published in 2006 has not been bettered. And, we're yet to hear about the one or two patients that have been treated by gene therapy from, two outfits, probably three outfits, And then we've got Dan talking about the placebo business and others, quite right, about it being unethical. I think someone was saying that earlier. I think it may be Nina. The Amethyst trial is a 2-year trial for patients over the age of 18 with GM2, where there's a compulsory 2-year arm of placebo in a 2-to-1 design, because neurological diseases that are like that are very difficult to work out. And when it's approved, the regulators will say, we need to be confident that there was a real effect. So, it's become a very tough world. And even Dan and his colleagues, and I'm sure NTSAD and the company at the time, Sanofi, could not persuade either the FDA or the EMA that the placebo-controlled arm double-blind crossover later has to be taken out, is unethical. And yet, so, you know, that's a tough call in a neurological disease, as you point out. We didn't get through on that, you know. So we don't have much of a veto.
I think it would be interesting to maybe hear from Ruben and Nina on this, because this is
Yes.
two people quite passionate about potentially trying to get studies going minus placebo arms. Everyone knows my view on that, but we need to avoid that if possible. So maybe Ruben, you want to go
Ruben
first.
too.
Oh, Nina. No, absolutely, I cannot. Nina first. Ladies first.
Well, I think you bring up a great point. One of the things that we see is that many of our partners and clients are more and more looking towards the PRO, validated instruments.
Patient-reported outcomes.
Yes.
What the patients feel, quality of life,
Yes,
so on. So
exactly. And
pain,
a lot of them
movement.
are starting to see the need of developing or investigating their own disease-specific instruments. And we actually, you know, have a partner, Quality Metric, who is specialized in this area. They're known for their SF-36 quality of life survey, as well as many others. And they're really working, you know, they have the population data. They also are working closely with us to leverage the data that will be collected in the registry to validate instruments for various disease areas. So
now you'll have to be helpful for me, Nina, because I don't understand leveraging data for instruments. Could you be a bit more colloquial for me, please?
Yeah, so it's more about, so we can create instruments for a very specific disease area, but to validate it, so it's psychometric requirements and all of that, they need to take the data that's been collected in order to say, hey, this instrument will provide the evidence that the FDA requires.
But forgive me on this, and I'm going to leave it in a minute, but I need to be a bit argumentative. Look, that panel on the FDA is people, right? And we talk about this wonderful word, validation, okay? But what's valid is human feeling. You've said that. It's quality of life, it's human feeling, it's what the PROs, okay? So how do you validate that, right? Is it passion? Do you validate it by sheer, you know, force of argument? Or what data do you bring to the validation? They live longer, or they live with a smile on their face? You know, what are you saying? I want to know what validation means.
No, this is a big thing. Let me, if I step in on this one, Nina, just if you forgive me. It's just that
please do.
I think
it's your métier.
It's the big issue that we have is the regulators need to, I think, wake up a bit that the world has moved on. Even the last 5 years, things are different. They need to realize that what was important 5-10 years ago isn't so important now. And the way things are done have changed dramatically. And, you know, we can't be looking at doing placebos. We all know, for example, scientifically, it's the best thing. No one's going to argue that. But when you're the parent of a child that's dying, who is then given the placebo, they'll know. And they'll know because the way the world has moved by communication, social media, everyone virtually communicates like we are today. 10-15 years ago, people didn't communicate in other countries other than by phone. Now we use these social media tools. So you compare notes. And I think it's naive of the regulators to tell companies, families can't communicate from a clinical study. Yeah, you tell them that, they'll nod because they want to get enrolled into a study. But then we all know they're going to post things on social media and talk to other people in the study, and they can compare notes. And if your child is going down while the one next to your child is stabilizing, it doesn't take a genius to work out potentially your child's on a placebo. I'm not saying they are, but potentially that's the decision you'll make. Then you'll withdraw from the study. And then the problem you get is then the recruitment doesn't happen because people will shift to another study. And this is, I think, something that's going to need to be addressed very soon.
Very much a problem.
Globally.
We have it at the moment. And you could, the problem is that when someone is withdrawn, finding out whether they're actually on the active drug or not is concealed. The companies are not obliged to let you know unless there are particular reasons for this. We have it at the moment, actually. And it would be very helpful, indeed. Yes, and of course, you could always argue that in a sense it's calling the bluff. That if the family believe that the patient their child. Let's take it's a child or a young juvenile person, you know, for want of a better word, and that child is deteriorating. You've then got to have an element of trust between the person who's medically involved, because it has to be ethical and legal, but also the parents. And then there would be an iterative dialogue and some kind of contractual element that you could say, okay, I'm prepared to tell you that I believe our child, our relative, the person we care for, is on the placebo, and we're going to call that bluff. And you could indeed find a mechanism, and this sounds very fanciful, but it's just a thought. You could find a mechanism for re-entry at a certain point. And it's a bit of a be-all and end-all otherwise, but it does certainly call the bluff, provided the patients are scrupulous and the parents don't find the trial too exhausting, and then they may have an incentive to withdraw. There are all sorts of things that could go wrong, but there are ways for being cognizant of that and conducting a much more human, realistic attitude to people who are frankly equals rather than some subject or other who's very lucky to be on a trial. And there's a power relationship which is not always attractive, I have to say, between the regulators, the companies who sometimes invoke the regulators for the purpose of perhaps making their lives easier. Shall I put it like that?
Maybe that's a good one to move on to Ruben now, potentially, to get your views on this, if you don't mind.
Oh, thank you. And as always, Tim words are very inspiring, and this point that Tim has raised probably would deserve hours, if not days, of discussion, because from a methodology perspective, I could not agree more with Tim that the placebo harm is the only right way to do things properly. I'm not the person who can discuss about ethical aspects. We all say that would be unethical. From a purely pragmatic, and I would wonder how, if parents would be open to a study, to allow their child to participate to a study, knowing that there is a risk to get into a, let me say, not active. The activity of the drug must be established, but this is certainly something that's important. The message, I have to say, that I bring back home, and I already transmitted to some colleagues of mine through the telephone, is that there are several companies, in this field at the moment. I would say almost incredible if you think about it. But still, and there is lots of expectation in this disease, of course. But I see that things are not so easy to move forward. The question would be, how could we work all together? And the point is that we cannot think of a relationship between the single company and the patient communities and the patient advocacy group, and a one-to-one, but we should truly work together. And the role of the academy is central. Is the triad, is a triangle. It's not like a line going into different directions. How could we, how could we increase participation to trials? And also looking at what Jennifer and and Jen were sharing with us, which is important. I was expecting the gene therapy trials to be so exciting that the enrollment could have been much more rapid. And I guess that is driven by logistics reasons. Certainly everybody's interested into the gene therapy. If I would have a child with the disease, I would go around looking for a center where he or she can get the investigational drug. So how can we work for that? It's it's a mixing of different things. And I avoid to comment on the health authorities because I had burning experience in the past, especially in rare diseases, where, especially when you go to the US authorities, there are some dogmas, or maybe we call it precedents, that they don't want to go beyond that. always tell the story that with miglustat, when in 2010, after an advisory committee which gave a positive vote, the FDA decided not to give us approval because the p-value was 0.056. And when we went to discuss with the FDA, somebody, I don't say who, said, why don't you do a prevention study in siblings of patients who have already clinical manifestations of the disease? And my answer was, well, madam, if you give us 80 years, 80, not 18, 80 years to run the study, probably we're going to be able to do it. So it's a mix of different things. And
yes.
And Tim, we need to all together to go beyond. And I'm not saying it because
no,
it's correct to say that. I think we need to find a way, share resources, share investments from a company side, and try to understand better. There is no competition in science.
Oh, interesting. I've got that's interesting. It leads me to a question for Jennifer. There are a lot of the Jennifer Sayo, that was because of the two. The conversation today, a lot's been on the advocacy involvement. I think I've been a parent, you've got the patients and the advocacy groups. What more can we drive things forward? I think as individuals, that's what we want to know. And we're really pleased you get us involved, but what more can we do to help?
Yeah, well, thanks for that question. And I think going back to what Ruben said about collaboration between the companies, I think really the role you can help provide is, a forum for bringing different companies together to collaborate, whether that be, generating natural history study data and finding a way to share that across companies, or working on initiatives for earlier diagnosis to try to decrease, the diagnostic odyssey. I mean, that's good for families, but it's also good for us running trials because we want to enroll patients early in their disease as possible, as well as getting things in place for newborn screening, which will be critical once these therapies are improved. So I think anything you can do to facilitate bringing us together to collaborate and share. I think speaking with many of my colleagues here, that's something that we're all very supportive of, and know we need to do the right thing for the families.
Could I ask you, Jennifer, sorry to talk a lot, but were you able with your trial design, which is semi-familiar, by the way, were you able to go along with patients or their carers as advocates to the FDA? Is that allowed? Actually, to go into the hearings, with the pre-IND and the IND hearings, yeah.
The FDA, I joined the company about a year ago, so I'm not sure for those meetings, but I know moving forward, we're working very closely with, we've put together a patient advisory council, and we're involving, those caregivers in a lot of these interactions.
I wasn't trying to trip you up, but all I can say is I've been to both places, but only pre-IND for the FDA, and biomarkers and so on. But for the EMA, several times at all levels I've been, and on each occasion it was possible you had an informed, not hysterical, an informed, organized, very thoughtful, very connected, one or two patient advocates at each level, one for the company, one for the, at all levels of the debating organization, and it was so compelling at each time. Also, in the UK, we have, just because I don't want to be Little England, it's embarrassing, but we do have something called NICE, which is a tertiary regulatory thing for actually whether the National Health Service will pay for a drug. That's very rigorous. A third tier, and there it's been absolutely essential to have patients with us who've had experience of the drug. But that's just by the by. Those things are coming in, I think, in other places too. But yeah, anyway, the partnership has been very real on registries and cohort studies and real cohort studies for the real world, which and going to regulators has been pretty good. But as I say, in the end, with the Amethyst trial, we failed to prevent the insistence of a two-year, which is a long time, placebo-blinded control trial. Yeah.
Yeah, I know. I absolutely agree with you. I think it's critical that we, as we communicate with the FDA and other regulatory authorities and have meetings with them, that we actually bring the voice of the patient organization with us, whether it be through live or virtual interactions or through some of their insights that they've shared.
Yeah.
The question I want to answer now actually is to Jen from Tay-Sachs. Is that we've talked regularly about finding patients, how difficult that can be, and we had the talks yesterday that focused on newborn screening. Do you see this as an initiative we should be trying to do globally rather than just kind of nationally in each country?
I do think that coming together globally to work on newborn screening is very important, especially if, there might be sibling data that is collected in some of these trials that can be, shared through the different regulatory newborn screening processes. I know in the U.S. with the RUSP application, ideally it's, great to have the data from the U.S. patients, but they do also look at information that is outside the U.S. as well. So I think those conversations being incorporated into, for Dan, I know we've talked about UK newborn screening and being part of the process, and to kind of work together with the findings that goes into the application, I think would be very helpful globally.
Mm-hmm. Well, thanks, Jen. That's obviously something we're keen on doing. I know I've discussed it with a few other patient groups to work out a way to do it. I mean, there's lots of stakeholders, lots of challenges, lots of hurdles. But to be honest, I think if no one does it or even tries to do it, it will just never happen. And we just need to come together to try and enforce that change.
And if you have those resources available, with everybody on a global scale, then there's less time spent to track down information. If the UK is looking for something particular that one of the other countries may have, to be able to support the effort.
Mm-hmm. Well, thank you, Jen.
Reduce the time anywhere you can. Try to do.
So I thought we'll just finish up this panel. I thought if I could ask all of you just your views, very quickly, just in like a sentence or two, I look at the last 10 years, so when we got into this, where we had nothing really happening, no company involvement. We had Tim, obviously, who I know very well, know what he'd been doing, but nothing really coming into the clinic. 10 years later, we now have all of you guys from different companies presenting at our conferences. It's a huge, huge shift. Where do you see us in the next 10 years as a community? It'd be good if you could have a sentence or two on that. Tim, I'll start on you because I know you've always got an opinion on this.
Well, I think you're essential, and even more so now, to give a bit of realness, as it were, reality to things. But I think, and keep all the companies and all of us on our toes. We have to step up when we talk to you, in front of you. You're informed and you ask the right questions. For me, I think the thing you can do beyond your existing registry of about 110 patients throughout Europe, at the last count, which is very valuable as it is, I think the thing you can do is to really give the guidance in the different communities about testing and prenatal and postnatal diagnosis, adding it to the panels. In Europe and the UK, it's very conservative. In the States, terrible mistakes have been made, for example, with Crabbe, which was, has so far been extremely difficult. Where detection did not lead to a clinical benefit with a bone marrow transplantation service and so on and so forth, all sorts of things happening. So making sure that the screening is better and that once therapies are there, that the centers are in place to deliver them and the experts to share them and keep the expertise up in front of you. I think before me, in front of you, and no doubt NTSAD, which I also know very well, though changing at the moment with change of CEO, is incredibly important. We all step up to behave. That's what I would say.
Great. I'll move on to Lisa. Do you want to go next?
Sure. 10 years from now, I think we all have some commitments we need to make to be in a better place 10 years from now. But my final thought would be that if something works even a little bit in the populations that we can get into studies, then as Jennifer mentioned, we see the diagnostic paradigm shift. Yeah. We identify these kids earlier, and we may find out that we actually have some great therapies out here if we can get them to babies at an earlier point in their disease trajectory. So I'm very hopeful about 10 years from now that there are multiple efforts in clinical trials.
We'll move on to Ruben.
What Lisa just said, I think, is extremely important. Forgive me for, I mean, maybe we have expectations which are unreachable. I think let's be happy of the step-by-step approach. To move forward, to do the first step for a long walk, because to me, and this goes back to the health authorities' point, well, maybe gene therapy has the potential to become a cure, but I mean, we are not targeting a cure at the moment. I mean, unless the only cure would be gene therapy. So let's be proactive but modest at the same time, and work together, truly all together for the next step. Because otherwise, it's a circular argument. Tim, I don't know if you.
I think that's brilliant. Yes, I do. I take both your points. I think the stepping up each time, if there's something, it changes things enormously. I mean, we have one trial in phase 2, phase 1-2 at the moment. We have several phase 2. We have the acetyl leucine and so on, derivatives and so on. So, yeah. It's not going to be curative, but it's appearing to offer things already, and it makes a very big difference for, as you said, Lynn, for referral practices, and it steps up. It becomes imperative. When I started in Gaucher disease, the expert had one patient. Now we've got centers everywhere. We've got a choice of 3 enzymes technically worldwide that are approved, and we've got 2 substrate reduction therapies, and we've got a trial of another substrate reduction therapy, and we've got a trial of a chaperone somewhere going on, and we're reaching out to Taiwan and Asia and everything, and we're even looking at the untreated in Pakistan maybe at the moment, because that's where we think patient delivery should occur. So you're right, it steps up and up and up. Yeah.
Thank you. Maybe move on to Jennifer from SIRE.
Yes, I'm going to try to be optimistic and hope 10 years from now we're having discussions about which treatment option is best for your child instead of not having any.
Yeah.
So
very good.
Very helpful.
Very good. Very good.
Let's move on to Nina, if that's okay.
Sure. So 10 years from now, I think with COVID, we started shifting the paradigm. Companies started embracing new approaches, new technology. And so what I'd like to see 10 years from now, and I'll echo what Jennifer said, is that we'll be talking about treatment, and we won't be talking about, breaking down silos, that we have all embraced this collaborative and innovative approach to identifying new treatments.
Thanks, Nina. And Jen from Tay-Sachs.
And then to add on, everyone's had such poignant pieces of information that, what I hope to see in 10 years is definitely a shift in the symptoms and the course of the disease. And so maybe just even talking about little baby steps in terms of better milestones hit and shifting the natural history of the disease course would be something I'd like to see in 10 years.
Of course. Yeah.
That's wonderful.
Society is greedy, of course, but that's real, isn't it? Yeah.
Cool.
Yeah.
Well, if no one else has any more comment, everyone's given their view. Double check. Missed anyone out. I'll welcome in Beer, who's going to do some closing comments, and we also have the book reading that will follow afterwards. So I want to say thank you very much to all of our panelists for your insights today, presentations. It was absolutely fantastic for our families to hear from all of you. And I know everyone's very grateful you could make the time to join us today. So goodbye, and thank you once again.
Thank you, everyone, for joining us.
Thank you.
Well, this has been an interesting panel, Dan. I think that to finish it, to sum up, I think you and I and all the patient organizations have a long way ahead, full of work. And I think that one of the things we've learned from these 2 days of conference is that we need more collaboration. We've got the tools now for that global collaborations with technology, with sharing data, with locating more patients. So for sure, I think the future will be brighter than it was a few years ago.
No, for sure. I think what I've really realized today is that we're very fortunate in the advancements we've had in the last kind of 10 years, not just on the treatments, but in the way health information is being developed, how that's transformed the way we can all communicate. The fact we can do these events virtually, which, maybe even 2 or 3 years ago would have seemed impossible for us to do because of technology. I think we're all now much more willing to try new things. And I think it's the same with the companies. They're all excited to try new ways to reach patient communities, use new drugs. They've got these amazing platforms trialing out drug matching, which I think is fantastic and will lead not just for us in the GM2 space, for other diseases out there, the opportunity to have future treatments. And I think that's a really exciting thing for us to look at. And I really think moving forward, we're lucky that the companies involved want to bring us in as advocates and also as patients in the community. I think that's going to make a massive difference for us, to be honest. I think that we can share experiences, we can ensure that those clinical studies are put together with the patient at the forefront of the discussion. And that's a phrase that I've used many times, but I think more and more people are actually starting to do that. And I think, in the next 10 years, we could be looking at my kids actually having a future. And I think that, again, is something I'm really excited to be part of. So anything else you want to cover, Beer, before we invite Emma to do round 2 of the book reading?
We have discussed a lot of important issues, data, screening, genetics, and above all, we can see how far we have gone from the first conferences to this one, where we have had a lot of industry partners here really interested and really learning to the GM2 gangliosidosis community. Now we don't talk anymore about scientific research as an abstract idea, but we talk about what treatments are now having clinical trials. So that's a big step for our community. And let's go to the closing for the conference.
Thank you, Beer. So I bring in Emma to do the book reading. Hi, Emma.
Hi.
Hello.
Cool. So we're going to invite Emma to do round 2 of the book reading after some tech issue yesterday. So Beer and I are going to step away while Emma reads the book. And let me just get that open for you now. And this should work perfect for you this time. Thank you, Emma. You can begin.
Thanks, Dan. A present for my sister. Written by Rosalind Stopps and illustrated by Jessica Fitzhoward. For our sibling squad, you're all amazing. This is Rose. Rose loves singing, dancing, eating chips, and giving people presents. This is Rose's mum. She loves reading and having 5 minutes peace. This is Rose's dad. He loves gardening. This is Tom, Rose's baby brother. He likes emptying the washing basket, feeding crusts to the dog, crawling up the stairs, and keeping everyone awake at night. This is Sadie, Rose's big sister. Sadie likes being close to Rose and Tom and hearing them play. Sadie isn't very well. Sadie is poorly all the time. She doesn't get better like Rose does when she has a cold. She lies on the sofa or sits in her special chair. Sometimes Rose wishes that Sadie could play with her. Sadie has Tay-Sachs disease. She has had it since she was born. This makes Sadie's life very difficult. Sometimes people come to try and help her. Doctors, speech and language therapists, physiotherapists, wheelchair specialists. Rosie and Tom sometimes feel left out. Sometimes Rose and Tom feel very sorry for Sadie. Sadie can't sing or read or empty the washing basket. She can't even eat chips. Sadie's food goes straight into her tummy through a tube. Every person is made out of different parts, like a jigsaw puzzle. A very important piece of the jigsaw puzzle that makes up Sadie is missing. It has been missing since before she was born. Rose has looked for it everywhere, but Mummy and Daddy explained to her that it wasn't anywhere to be found. Sadie is how she is, and that can't change. Rose has an idea. She decides to give Sadie a present to cheer her up. She asks Tom what he thinks, but Tom speaks baby language. A jack-in-the-box would be too scary. A kitten would be too jumpy. A toy farm would have too many pieces. A doll's house would be too fiddly. A drum, useless if you can't bang it. A book is dull if you can't see it. A doll isn't much fun if you can't carry it around. A car is no good if you can't vroom it along the floor. Rose thinks of everything she can, but nothing seems right. Rose asks her mum and dad what she should give Sadie. That's easy, says Mum. That's easy, says Dad. Mum and Dad explain that Sadie would like some of Rose's much more than any present she could give. Time with Sadie is the best thing you can give her, they say. Sadie can't play, but you can play for her, Mum and Dad say. She can't read, but you can read for her. Mum and Dad explain to Rose that Sadie won't always be with them. One day she will die, they say, because of the missing part that can't be mended. Rose asks what will happen to Sadie when she dies. Different people think different things, they say. No one knows for sure. Some people think she may go to heaven. Some think she may be asleep in a comfortable place where she doesn't have to wake up anymore. Maybe she will go up in a spaceship and visit the moon and the stars. Rose is very sad. But what's important is that you know she will always be looking down at you with a big smile on her face. Sadie will always be in our hearts, says Dad. You can always do things for her, like you play for her now. Rose decides to give Sadie a piece of her time every day while she can. She knows that Sadie will die, and that will be very sad, but she will always have her big sister in her heart.
Thank you, Emma, for a beautiful reading.
Thank you.
for that. So now the conference has now finished. We've just got a very short closing video, and we will be in touch. If you have any messages or questions, please send them to us via the platform or by email. And we hope to see you in person at the next event next year. So thank you very much, everyone, for attending. It's been fantastic.
It takes a spark